Science is not just a subject, it is the art of questioning the universe. Good morning, respected dignitaries, distinguished faculty, researchers, and dear visiting students. Today is not merely a date on calendar, it is a reminder that a beam of scattered light once redefined India's scientific identity. On this day, we celebrate the discovery of Raman effect by Sir C. V. Raman. But beyond celebration, Science Day is a reaffirmation of our commitment to scientific inquiry and translational innovation. It is with that spirit that I, Smith Kamble, have the privilege of welcoming you all on the behalf of Manipal Institute of Regenerative Medicine, MAHE, Bengaluru, for today's celebration. I would also like to extend a warm and special welcome to the students who have come from Christ University, Ramaiah College, and Karnataka Pharmacy College. To formally commence today's proceeding, I would like to invite Professor Jyoti Prasanna, Dean of MIRM, to deliver the welcome address.
Good morning, one and all. I think the Josh is alive, right? All of you had breakfast because we are going to have a very rigorous day, the first half, with nice scientific presentation, especially Professor Balaram's talk. She's going to talk about her many decades of scientific journey in the field of malaria parasitology. You should listen to her carefully and engage with her. She's a kind of scientist who likes interacting with students and tossing new ideas. Followed with that, we have scientific talks by our PhD students and the master students and the students at the back who have come from different colleges across Bangalore should benefit and know what we do at Manipal Institute of Regenerative Medicine. So every year we gather in this auditorium, generally on 28th February, but this time due to other commitment we had to postpone it for a week to commemorate the discovery of Raman effect. All of us know what the Raman effect is and how just a simple question of why the sky is blue led to the entire philosophy of how light scatters and how the light scatters. The information of how the light scatters can give information about biomolecules. It can give information about tissues because every particle has a unique character. It has a way by which it deflects light or scatters light, and that scatter spectrum can identify a molecule. And now, Raman's effect is not just limited to physics. It has touched all streams of biology and medical diagnosis. So this day is not just a day to celebrate Raman effect. It is a celebration of our collective scientific journey and the endeavor to keep the spirit of inquiry alive in us. So on this day, in general, every year we call an eminent scientist who talks about their scientific journey right from their PhD to how they started their lab and what stage they are in and how basic fundamental research had led to innovations in the field of healthcare. On the day, we also celebrate our small accomplishments at MIRM, where we unveil our newsletter, Rejuven, where we have contributions from students, we have contributions from faculty, and we brag a little bit about our accomplishment. It is very, very important to do that because the master students are going to be the thought leaders and opinion leaders tomorrow. So they should have an opinion about different concepts, whether it is geopolitics or it is science or innovations across the globe. So we celebrate that as a part of our newsletter. Followed by that, we have flash talks by PhD students from each lab where they have to give a gist of what they are doing in 10 minutes. And that itself is an art. Science has to be broken down in simple language. Then only you know your problem statement clearly, and that's why the timing is very, very limited. Today is particularly special because the theme of this year's Science Day is Women in Science, Catalysing Viksit Bharat. I would want to stand here maybe 5 or 10 years down the lane and say that we should not have a day where we have to celebrate women, because that should be organic, that when you're in a lab and when you're wearing a lab coat, it doesn't matter whether you're from a particular community or whether you're of a particular gender. I hope we should not celebrate it in the coming few days, but definitely proactive measures have to be taken to reach that point. So with this, I thought it would be best to invite an eminent woman researcher, Dr. Hemalatha Balaram, to speak about her scientific journey today. Her pioneering research on metabolism in Plasmodium falciparum and dedication to structural biology at JNCASR is very well known to all of us. She's going to talk about how her fundamental research in the field of Plasmodium biology has led to identification of drug targets in the field of malaria. So, as we strive towards Viksit Bharat, we are talking about Viksit Bharat. If you look at the census, which happened in 2023, I think, correct me if I'm wrong, the gender parity has been sort of bridged because, you know, you have almost similar numbers of males and females, thanks to proactive steps by the government to prevent female infanticide. But still there exists a gap. I was looking at the statistics yesterday as to why we need to have women in science, and I was very appalled looking at the numbers. If you look at the number of women who enroll for higher education in India, it is 43% in STEM. So that's quite a bit of a number. But if you look at the number of working scientists and faculty in STEM, it is a mere 16%. So from 43% who enroll for a STEM program in education, there is a decrease in the number of working scientists. More so, I don't know whether I should say this in this podium or not, but in elite institutes like the IITs and ISC, where I have done my PhD, the representation is only a mere 9 to 10%. In terms of, if you look at streams, if you talk about physics, biology, representation in physics, biology, mathematics, biology has a representation of 22%, physics stands at 16%, and mathematics, again, just 10%. So there is a mindset problem as to whether women can't do, or they are not given the opportunity to do them, or they are made to believe that some streams are not there for women. I think we need to talk about it. I don't want to talk as a feminist, but these statistics are a reality. And till now, if you talk about national awards like the Shanti Swarup Bhatnagar Awards, around 250 awards have been given to people across all streams. And out of them, the women fall in single-digit number. So you can clearly see that definitely the contributions of women in science are either not recognized, or they don't make up to that level, or they leave somewhere in between. And it is high time that we understand why this is happening. I don't want to sound too melancholic and say that women can't do or can do, but what is important is that we have a legacy from Curie's, the family of the Curie family, where they have actually shown this equality much, much long before, during the times of World War, where Marie Curie got 2 Nobel Prizes, one in the field of physics and one in the field of chemistry. And not just, she actually, even her daughter, who is Joliette Curie, had also got a Nobel Prize in 1935 for discovery of artificial radioactivity. So clearly, women are capable in an era where people were not talking about gender equality, right, and feminism. So the capability is there. But where is this lost is something which we need to talk about. So the mother-daughter duo getting the Nobel Prize is not just individual success. It's a generational shift. And that generational shift is what we have to incorporate in our psychic. So to all the young minds and the researchers who are present here, you are true catalysts of developing India. I want all of you to become opinion leaders. Talk about your thoughts unabashedly. Talk about research and science is not just about what you do in your labs and what you do in your Petri plates. It's also about having an opinion, engaging with the community, understanding the real world problems, and giving back to the society. Ultimately, I always keep talking about it on the lunch table with my faculty that we are using taxpayers' money. So everybody has a right and equity for resources in science and education. With this, I would want to implore all the youngsters here that today's discussion is not just to celebrate women or give them awards, but is to commit for a more inclusive and a sustainable future. When we talk about inclusivity, it doesn't restrict to only women, it restricts to geographical domains, it restricts to people who are differentially abled. And I would like to, when I was having a conversation with Dr. Hemalatha Balaram, she had talked that she's starting an initiative of educating students who are differentially abled, and how can we get them into science? Suppose somebody has a hearing disorder, but still they might be thinking their brain is fine. So how do we get them into science, into mainstream science, and what should be done? And I think only a woman who has that compassion and of that caliber can do it. I think we should commemorate Dr. Hemalatha Balaram for that thought process. Now I would like to request, without talking more, if you give me the podium, I'll speak forever. I think most of the students know that. Without wasting much time and handing over the podium to Dr. Balaram, I would request the chief guest, Professor Balaram, to formally start the program by lighting the lamp, which is followed by the digital unveiling of the newsletter, and then prize distribution by the meritorious students. Since we are celebrating women in science, I would request the women faculty of MIRM to come up to the stage and also come up for lighting the lamp. Please.
With the lamps now lit, may its light guide us towards knowledge, curiosity, and innovation. Thank you, respected dignitaries. Institutions grow through ideas, and when ideas gain permanence when they are documented. The MIRM newsletter, Rejuven, reflects the research, collaboration, and academic momentum that defines this institute. May I request Professor Hemalatha Balaram to join us on unveiling of our newsletter. Congratulations to everyone involved in bringing this edition to life. May it continue to capture the evolving stories of science at MIRM. Excellence deserves recognition, and today we honor the brightest minds among our students, whose dedication reflects the scientific pursuit. In a moment of well-deserved applause, let us celebrate our meritorious students. Firstly, we would like to recognize the toppers of MSc in Stem Cells, Technology and Regenerative Biology, batch of 2024. May I request Professor Hemalatha Balaram to kindly come forward and present the prizes. The first prize goes to Miss Evelyn Days Soni for securing the first rank. She is not here currently. We move on to the second prize, which goes to Miss Aishwarya S. Biradar for securing second rank. Next, we move on to the toppers of MSc in Translational Immunology. The first prize is awarded to Ms. Bhavya Shri Hada for securing the first rank. The second prize is awarded to Ms. Lavanya Yu for securing the second rank. Congratulations to our brilliant students. Let us have a big round of applause once again, please. We now recognize the toppers of MSc in Translational Neuroscience. The first prize goes to Ms. Karen Sneha P. for securing the first rank. The second prize goes to Mr. Osama Jamil Muhammad Mureb for securing the second rank. Osama is not with us currently. We move on to the next prizes. Finally, we will move to newsletter awards. May I now request Dr. Jyoti Prasanna, Dean of MIRM, to kindly present the prizes for the Best Newsletter Article Award. There are two prizes for this. May I request Meera Pradeep to kindly come forward on the stage to receive this award. Now I call upon Ms. Anuradha Krishnan on the stage to receive their award. Thank you, ma'am. A huge round of applause for all the prize winners and dignitaries for this gracious moment. Your achievement inspires us a lot. Keep questioning, keep discovering. I now kindly request all the dignitaries to take their seats in the audience as we proceed for the program. To elevate our celebration with profound insights from a visionary in science, I now invite our esteemed Dean, Professor Jyoti Prasanna, to introduce to today's keynote speaker. Ma'am, the stage is yours.
Yeah, I think ma'am can just sit on again coming up and yeah. So for most of us, Professor Hemalatha Balaram is not new. We are very well aware of her scientific journey. But for students, for just starting their career, I think it's very important to know about from where she has started and where is she now. Okay. So Professor Hemalatha Balaram is a J.C. Bose National Fellow and a honorary professor at the JNCASR Center in Bangalore. She received her doctorates from the Indian Institute of Science and did her postdoctoral research at the University of California, San Francisco, USA. After a short stint at Astra Research Center, India, she joined JNCSR as an assistant professor in 1996 and has been a professor since 2008. She was the Dean of Academic Affairs from 2008 to 2011 and the Dean of Faculty Affairs from 2015 to 2021. She is a resident honorary professor since November 2021. Professor Hemalatha has several years of experience in the areas of molecular parasitology, metabolism in parasitic protozoa, molecular enzymology, and protein structural functional analysis. The focus of research in her laboratory is on understanding biochemical pathways and metabolism in malaria parasite Plasmodium, as well as protein structural functional analysis of enzymes. So you can see a mix of probably understanding of metabolism, computational biology, drug design, all of them interspersed together. So you can see how innovations can happen only when we have an interdisciplinary focus. And science is actually field agnostic. I think her research journey is a testament of that. She has more than 100 peer-reviewed publications. She's a fellow of the Indian National Science Academy and the Indian Academy of Sciences. She's the recipient of Dr. Raja Ramana Award for Scientist, Karnataka State Council for Science and Technology, and the CNR Oration Award. She has served as a convener, member of various task forces and sectional committees of different government bodies, and has a strong opinion and is a strong scientific voice to reckon with. She was the president of the Society of Biological Chemists and is currently the chair of the science education program of the science academics. And I would just want to talk about it. We were having a conversation in the morning and she said that SPC has been conceptualized for more than 100 years, right? And she, yeah, in 2030 we'll be reaching 100 years and there is only one female chairperson in all these years, and that was Hemalatha Balaram. So I think she had a voice and she made sure that the voice matters, not just scientifically, but also as an opinion leader championing for women equity. I invite you for your presentation, Dr. Bharat.
and their contributions to developing India and taking it forward. I've just put here 3 Indian women scientists who have made very fundamental contributions. This was all long ago, and they are no more. One is Dr. Ashima Chatterjee, who was a natural product and an organic chemist. It's from natural products that you can actually develop novel molecules for various diseases. She worked on anti-epileptic drugs, anti-malarial drugs, both from synthetic organic chemistry perspective and also from natural product chemistry. Kamala Sohoni actually joined the Indian Institute of Science, and in those days, as a woman, she was not allowed to do her PhD, and Kamala Sohoni actually went on hunger strike apparently and got her master's degree from the Indian Institute of Science in the area of biochemistry, and thereafter she went to UK and worked on cytochrome C, for which she got her PhD, came back to India and worked on various aspects of nutrition, vitamins, which were hot topics in those days. The third Indian woman scientist is Janaki Ammal, who did a lot of work in botany, especially in the area of plant breeding, cytogenetics, and phytogeography. The fourth person I have put is a Chinese lady who got the Nobel Prize for the discovery of the drug artemisinin. Before chloroquine was discovered, the treatment for malaria was using quinine. And thereafter, during the Second World War, there was a strong push to develop a drug very fast, and chloroquine came out. Very soon, the parasite developed resistance, and different parts of the world had chloroquine resistance. And during the Vietnam War, the Chinese decided that they would help and try and see if they can get a new drug for malaria, because a lot of soldiers in forest regions of Vietnam were succumbing to malaria. Yu Tu discovered artemisinin, and that story itself, I don't have time to tell you. You can read her Nobel lecture. It's a very interesting story of how it came out of Chinese traditional medicine, this molecule that she identified. Next, I've put Gertrude Alien. Yu Tu had only a bachelor's degree, and there was no concept of a master's degree in China in those days. This was during the Cultural Revolution when Mao Zedong was at his peak. So she had only a bachelor's degree. She did not go abroad for her, any further training, nor was she a member of any of the science academies in China, but she made the discovery. Next is Gertrude Alien. Gertrude Alien struggled to get a master's degree, and after master's degree, she worked as an assistant to Fred Hutchings, and there they developed innumerable drugs. The first, mercaptopurine, which is used for treating leukemia, including her contributions to AZT, which is now used for treating HIV, and numerous other drugs, including certain anti-malarial drugs like pyrimethamine, which is still in use, she developed. She developed actually almost 8 to 10 drugs before even the enzymes that make these molecules catalyze reactions in the cell were known. But they knew the biochemistry, they knew all the molecules that were there, but they didn't know the nature of the enzymes catalyzing the reactions. But she developed innumerable drugs and got the Nobel Prize in 1988. Put here are, again, 4 women. You must have come across the Michaelis-Menten equation in your biochemistry. Menten is a woman, that's Maud Menten. And Maud Menten is a Canadian who went over to Germany to work with Michaelis, and that's where they came across, she developed this, their analysis of enzyme kinetics led to the development of this equation, which is there in every biochemistry textbooks, and it is the fundamental basis for understanding catalysis carried out by enzymes. Then Rosalind Franklin, all of you all know of photograph 51. Photograph 51 became the basis for proposing the double helical structure of DNA. Dorothy Hodgkin, who first solved the structure of penicillin and vitamin B12 and insulin, remarkable contributions and over a range of molecules, again got the Nobel Prize. Isabella Karl, who again made very fundamental contributions to the way we do crystallography today, but did not get the Nobel Prize. These are the women who broke many, many barriers to come forward and do science, not in the kind of environment you have today here, or in institutions in India, elsewhere in the world, but the conditions where women were not encouraged and they were thought that one doesn't need to give them a stable job in an educational institution. So as Dr. Jyoti Prasanna pointed out, across the world, less than 30% researchers worldwide are women. And if you go up the ladder, the hierarchy ladder, you will find that the number who are there diminishes down to just 10%, very small, sometimes even lower than 10%. How can this exclusion be reversed? Itself is a subject that one has to have discussion on and find out ways by which you find ways to include more women at higher levels, decision-making levels, and an opportunity so that they can also contribute. You have 50% of the world is women. So if you just ignore them, what's going to happen is you'll ignore the creative power of 50% of the world. Yeah, so there is a need for gender equality, which is a fundamental human right. As I said, 50% are women and they have equal creative abilities. So to build a sustainable, peaceful, and prosperous world, you need gender equality. And the major thing that gender equality does is it breaks down harmful stereotypes and allows individuals to reach their full potential. So with this brief talk on what was the Raman effect, why did Raman get the Nobel Prize, and why is the theme today Women in Science for having Viksit Bharat, for having a developed country, I'll go forward into an aspect of the study that we carried out in my lab. So the talk is going to be on TCA cycle associated metabolism in the parasite. And I know most of you all don't like to study metabolism because you think that it's all to do with just memorizing. That's not the way. You have to look at metabolism from two perspectives. How does cell do chemistry? I'm a chemist. I am trained as a chemist. We do chemistry. If I want to make a molecule, the way I make the molecule as a chemist in a lab is completely different from the way the cell does it. The cell does it at ambient pH, ambient temperature. Everything is very, very ambient. We don't boil things. We don't add acids, strong bases, nothing. So the cells do it in a very simple manner. And the way of looking at metabolism is from two perspectives. One is, what are the kind of chemistries that are there in a cell, and how does the cell carry out these chemistries? The second is to look at metabolism from an evolutionary perspective, because you will find that there are fantastic deviations in certain organisms. Yeah, so Plasmodium, malaria is caused by Plasmodium, the parasite Plasmodium. We have over 250 species, identified species of Plasmodium. There is a vertebrate host and an invertebrate host. The invertebrate host is the mosquito, and the vertebrate host can be all primates, including humans, rodents, mice and rats, bats, birds, and reptiles. Yeah, these are the drugs that we have. Artemisinin is there right at the bottom over there. It's a structure with an endoperoxide OO linkage. That's why the discovery, the story of getting this molecule out from Artemisia, or sweet wormwood, is a fantastic story. To begin with, it was quinine, followed by chloroquine, and we have drug resistance to chloroquine. We have drug resistance to almost everything that I have put over here. We now have a new drug whose picture I haven't put here, which has been just recently, just a year ago, papers have come out, which has been cleared by FDA, and it is approved by WHO, and it can be used now in extreme cases of drug resistance. Otherwise, it's artemisinin combination therapy, artemisinin with pyrimethamine and sulfadoxine, or artemisinin with doxycycline and sulfadoxine. So these are the combination therapies that one uses. And the major problem is drug resistance, as I told you, and the fact that we don't have an effective vaccine. So if you look at the life cycle itself, I have put of Plasmodium parasites, which infect human beings, on the left, and on the right you have life cycle of the parasite Plasmodium berghei, which infects rodents. I work on both Berghii is genetically tractable more easily than falciparum. And in Berghii, I can go into the mosquito stages. So here, you can see there's a mosquito that's biting a human being, and if the mosquito is infected with a parasite, along with its saliva, it injects a stage of the parasite known as sporozoids into your bloodstream. And from the bloodstream, it goes first to the liver, where it undergoes hepatic schizogony, that is, invades liver cells, multiplies, and one mother cell gives rise to innumerable daughter cells, which burst from the liver cells and enter your bloodstream. In the bloodstream, they survive within erythrocytes. So it invades erythrocytes, and one mother cell every 48 hours, in the case of Plasmodium falciparum, gives about 35 to 40 daughter cells. And every 48 hours, when it ruptures and releases its daughter cells into the bloodstream, that's when you have fever and chills. And that is when the disease itself is manifested. During the liver stages, disease is not manifested. Some of these asexual parasites within the erythrocyte differentiate into gametocytes. And if the mosquito now feeds on this infected individuals, it sucks up the gametocytes, it sucks up blood, and also along with it some level of gametocytes, which inside the mosquito, they develop into gametes. Gametocytes become gametes. The gametes fertilize, they form an zygote, then an ookinete, and then they form sporozoids. They travel up to the salivary gland and form sporozoids. What I can study in the lab, if I am working with Plasmodium falciparum, is the erythrocytic stage itself, which I can culture using human blood. If I'm studying Plasmodium berghii, I can study the erythrocytic stages, I can study the gametocytes, I can actually study all the way up to ookinetes in the laboratory. Those are the mosquito stages. Then that tells me how does interfering with certain genes involved in metabolism hamper development of all the stages or any one of these stages. Yeah. So when the genome sequence was published way back in 2002, there was an idea of what are metabolic pathways, and many of the drugs that I showed you interfere with key metabolic processes, and those are the drugs that we use right now. So this is the picture that I've taken from the paper in 2002. So Plasmodium is a microaerophile. It needs low level of oxygen and not the level, so we grow it under low levels of oxygen in the laboratory when we grow the parasites. During the intraerythrocytic stages, when the disease itself is manifested in human beings, it produces all its ATP primarily through glycolysis. It doesn't seem to depend on the TCA cycle and the electron transport chain for ATP production. It has other features too. It has no amino acid biosynthesis. Every amino acid is taken from the host. It's either from the human host or it is the mosquito host. It doesn't have a pyrimidine salvage. It cannot salvage pyrimidines, but it also doesn't have a de novo purine biosynthetic pathway, which we humans and most free living organisms have. So since I'm going to talk about the TCA cycle, I'll tell you a little bit of history about the TCA cycle itself. It was Hans Krebs who discovered the TCA cycle. And Krebs, when he was originally working in Germany, and at the height of the Second World War, he moves out of Germany. And the way he did these experiments was to take tissue, rabbit muscle tissue, and see how much oxygen is consumed based on what he was feeding. He would feed different organic molecules, like intermediates of the TCA cycle, sugar derivatives, and so on and so forth, and see how much oxygen is conserved. And the equipment that he used was a manometer to see how much oxygen has been consumed. And this is the cycle as we know today. And the original paper itself was published in the journal Enzymologia, which is no longer there. And this is the classic paper by Krebs and Johnson that shaped all of modern biochemistry. So before Krebs came into picture, Martius and Knoop had put down the reactions of the TCA cycle, but they put it down as a single step of reactions. But when Krebs was doing these experiments using the manometer, what he realized was He had already gotten the urea cycle. But that didn't influence him. But when he was doing these experiments, he realized that these levels of these organic acids does not change even if it is being consumed. So if it's a linear pathway, yeah, the levels will change. But if it's a cyclic pathway which regenerates it again and again, you will not see a difference in the quantity of the organic acid that you have put on the tissue. So he decided that it has to be a cycle, but he didn't know what collected the cycle at that time. It was subsequently found out. Yeah. The cycle doesn't operate in isolation. The cycle can have intermediates being taken out of the cycle, catapleuroses that is called, and intermediates that can feed into the cycle, which is anapleurosis. And the term anapleurosis was actually given by Hans Kornberg, a biochemist in UK, and I was looking at where did he get the word anapleurosis. I mean, it's not a common English term at all. So in his obituary, written in the journal Guardian, they say that with a love for classicals, that is his classic literature, derived from his grammar school education, he called these anapleurotic reactions. It is a word derived from Greek, to fill up. Anapleurosis means fill up. So when you're having TCA cycle intermediates going into the cycle, you're actually filling up the cycle. Anapleurosis is a widely relevant concept in biochemistry. Why am I talking about TCA cycle and anapleurosis? Years ago, we were looking at, I told you there is no purine de novo biosynthesis, purine nucleotide de novo biosynthesis in all parasitic protozoa, not just Plasmodium. All of them salvage precursors from their host and convert them into the nucleotides it needs. What are the two key purine nucleotides that a cell will need? AMP and GMP. Those are the two key nucleotides that the cell will need. The way the parasite produces, Plasmodium produces AMP, it produces a molecule of fumaric acid along with every molecule of AMP produced. And as I told you, it was thought that the TCA cycle is not important. Fumaric acid is an intermediate in the TCA cycle. And a question that we asked some years ago, my student Vinay Bolusu, was what is the fate of this fumaric acid? If the parasite doesn't have an active flux through the TCA cycle, is it just a metabolic waste? The parasite also has what is drawn on the right over there, what is called a purine nucleotide cycle that starts with the purine inosine monophosphate, abbreviated IMP, gets converted to succinyl AMP using a molecule of aspartic acid, and the succinyl AMP is now hydrolyzed to give you AMP. And there it produces fumaric acid. But the cycle can go, AMP can get converted back into IMP, and if the flux through the cycle is high, you produce a large amount of fumaric acid. And in exercising muscles, this purine nucleotide cycle becomes important because all the fumaric acid that is produced goes into the TCA cycle, and thereby it will go into the produce reducing equivalents. The reducing equivalents go into the electron transport chain and produce ATP that is needed for an active system. Parasite has this purine nucleotide cycle. So it does produce fair amount of fumaric acid. The question was, when the flux through the TCA cycle is very, very low, is fumaric acid a metabolic waste? What is the fate of this TCA cycle intermediate? So that's the question. There is the purine nucleotide cycle producing fumaric acid, and the parasite divides every 48 hours. Its requirement for purine nucleotides is very high because it has got to synthesize DNA heavily. So this fumaric acid in our system flows into the TCA cycle. Is there an anapleurosis of fumarate in the parasite? And what is the significance of this anapleurosis? So the first experiment that we did long ago was to put labeled fumaric acid, and that's the structure of fumaric acid that's shown in the middle over there. And there are two carbons with a star on them. Those are both C14. C14 carbon is radioactive. And what we found was, surprisingly, from labeled fumaric acid, we got labeled DNA, we got labeled RNA. We also got labeled proteins as shown in the right panel over there. That's an autoradiogram showing labeled proteins. So C14 derived fumaric acid incorporates into DNA, it incorporates into proteins, and in DNA, it labels the pyrimidines only and not the purines. That's what we found out. Next, what is the fate? It's a small metabolite. So it has got to be metabolized, converted into other metabolites. And how do I monitor that? Way back when we did have good NMR machines. And as a PhD student, I was trained in nuclear magnetic resonance spectroscopy. And I was familiar with the use of nuclear magnetic resonance spectroscopy. And we put here not C14 labeled fumaric acid, we put C13. C13 carbon is NMR sensitive. And when I put that, I could now see peaks coming up. They're all called chemical shifts. And these chemical shifts are very characteristic of a carbon in a particular metabolite. It's a fingerprint of the molecule. So I saw that fumaric acid there is malic acid that's produced, there's lactate that is produced, there's aspartate that is produced, and there's pyruvate that's produced. So I can now go back. I know metabolism. I can go back and track what is getting converted from one to the other. And when I track it, I find that fumaric acid to be converted to malic acid, I need the enzyme fumarate hydratase. And then to proceed further, I have put every enzyme that I need. And today's talk, I'm going to talk only about the enzyme fumarate hydratase, which converts fumaric acid to malic acid. And the first thing that we did was, since it's a TCA cycle intermediate, where is fumarate hydratase localized? TCA cycle happens in eukaryotes within the mitochondrion. So the question was, does the protein localize, the protein which converts fumaric acid to malic acid, does it localize to the mitochondrion, or is it to both mitochondrion and cytosol? So we tagged the endogenous copy of fumarate hydratase with GFP, and there is the study which shows the yellow color in the last panel that you have, in the bottom panel, that shows that this enzyme, which converts fumaric acid to malic acid, that is fumarate hydratase, which I'm going to talk about, localizes to the mitochondrion in Plasmodium falciparum. So begin with this information, we decided to do reverse genetics on the parasite, knock out genes, and I'm going to talk about knockout of many genes are there, but I'm going to talk about only of fumarate hydratase because I don't have time to cover all these. There we were trying to look at different aspects of TCA cycle linked metabolism. I'm going to talk only on fumarate hydratase today. And what am I going to look at the stages? I'm going to look at the erythrocytic stages shown there in red, that is starting with an uninfected red cell, the ring stage, trophozoite stage, and the Schizon stage. That's the erythrocyte cycle. I'm going to look at the gametocytes. I'm going to look at zygotes and ookinins. So I'm going to first look at the phenotype of the parasite as a consequence of this genetic, reverse genetic approach by which I knock out the FH gene. And then ask, after looking at the phenotype, ask what are the metabolic consequences. Phenotype, I look at it under the microscope. I know parasite morphology, and I look at the phenotype under the microscope. As I said, the parasite doesn't depend on the TCA cycle during its asexual erythrocytic stages. And shown on the top left panel, you can see between the wild type and the knockout, there is no difference in growth rate of the erythrocytic asexual stages. Gametocytes also do not show any difference. But formation of ookinins, where the gametocytes have to get converted to gamete, gamete to zygote, fertilize and form a zygote, zygote to ookinin, that's completely abolished. So it is after the gametocytes is formed, when it has to become gametes and transition, fertilize and transition into ookinins, that's completely blocked if I remove a TCA cycle enzyme, that is fumarate hydratase. So now I can take gametocytes and ask the question, that is the last stage which is alive. After that, I don't get anything. They're all dead. So I can ask the question, how is metabolism altered in these gametocytes which do not have the enzyme fumarate hydratase? Yeah, how do I do it? Today, I won't use a manometer. Today, I can use mass spectrometry because it's really advanced and you have resolution to the level of 5 ppm resolution that even two molecules which differ by a very small mass can be distinguished. So I can do it two ways. I can do it by doing a relative quantification of metabolite levels across wild type and the knockout. The second I can do is I can look at way metabolism, the flux itself happens in the knockout and in the wild type. For the first study, I just take parasites and look at metabolite levels. For the second study, I use a stable isotope, C13 labeled isotopes to follow metabolism. I'll show you how we do it. Yeah, that's the result first. This is just a comparative quantification of metabolites, and I won't tell you about all the metabolites. I have knocked out fumarate hydratase. That's the red cross there that is put. Fumarate is produced when AMP is synthesized. Fumarate will also get produced through the TCA cycle. And since I don't have fumarate hydratase, that is blocked over there. What do I see? You can see that the levels of fumarate have increased. That's expected because there is no enzyme to utilize fumarate hydratase. But interestingly, I have got glutathione, which has become, you know, glutathione is a reducing molecule. It's a tripeptide. That has got modified and has become succinated glutathione. I also have what I call succinated cysteine. And if I look at the glutathione, the bottom left panel, in the wild type, there is a huge amount of unmodified good glutathione that the cell needs to survive and keep its redox system balanced. In my knockout, I have very low, the levels of unmodified glutathione have dropped to 50%. 50% is modified by what is known as succination of glutathione. How did this happen? I've just knocked out fumarate hydratase, I've accumulated fumarate. How did it go and modify glutathione? Sorry. How did it modify glutathione? What is the TCA cycle intermediates like? There is one monocarboxylic acid there, I've written down. There are two tricarboxylic acids, and the rest are all dicarboxylic acids. Those are simple organic molecules. Starting with citric acid, you can see alpha-ketogluteric acid, you can see succinic acid, fumaric acid, malic acid, and oxaloacetic acid, small organic molecules. Nothing unique about them if you look at it from a biological perspective, but the chemist will say there is a difference. And what will the chemist say? He or she will say that there is only one of these acids, organic acids, which has got a double bond. And which one? That is fumaric acid, shown there with red arrows over there. It's got a single double bond. And on either side, two COOH groups, that's carboxylate groups. Such a molecule, in chemistry, I would call it alpha-beta unsaturated dicarboxylic acid. Unsaturated because there's a double bond over there. Okay, that molecule can, without any enzymatic help, react with elements which are electron-rich. What is electron-rich in your system? The thiol, SH groups in glutathione, in free cysteine, and SH groups in proteins are all electron-rich atoms. And fumaric acid can undergo, in chemistry, what we call a Michael addition and modify cysteine. It can modify glutathione. And many years ago, few years ago, while we were doing this work, there were many papers which came up in the literature. There was a flurry of papers in which fumaric acid now was called an oncometabolite. And I'll tell you why. It's called an oncometabolite, and fumarate hydratase deficiency in humans is one of the leading causes for renal cancer because it modifies key proteins, cysteines in key proteins, affecting their function, leading to cancer, malignancy. So I've listed a few papers there, starting from the first one, which was identified as fumarate as an oncometabolite because fumarate can catalyze a reaction, can carry out a reaction which is known as Michael addition, by which it modifies the cysteine. So shown here is just cysteine reacting with fumaric acid, and what you have is the thiol of the cysteine forming an adduct with fumaric acid. Shown here at the bottom again is glutathione reacting with fumaric acid. The thiol in glutathione now getting modified into succinated glutathione. That is exactly what is happening in our system. In Plasmodium falciparum, it's depleting the reduced glutathione that is absolutely essential for maintaining the redox state of the cell. And we did the entire proteome analysis and tried to look for proteins which are succinated. And you can see here I've shown a subset of proteins which are succinated, not succinated in the wild type, but in the knockout, they get succinated. Specific 16 residues in the protein getting succinated. I won't go into this further, but we find that this happens. Just like in other cell types where this happens when you have fumarate hydratase deficiency, where this modification of thiols happens, and the reason why fumaric acid is called an oncometabolite. The second aspect that we study is I've seen this, I've done a comparative metabolomics, and I've seen fumarate levels increase, which is expected, and I have seen that this increase in level brings about a chemical reaction in the cell by which cysteine residues get modified. And the key thing that we see is that free glutathione levels drop because I don't have the cysteine now available for maintaining the redox state in the cell. The second aspect that I can look at is how is the flux changing. If I have a traffic jam at some point, you will have traffic flowing through another route. Similarly, if I block a pathway in the cell, you will have reorganization of the pathways by which you will have some chemistry happening through a different pathway. That's what I've shown over there in that. I can now look at this as a consequence of knockout of fumarate hydratase by using isotope tracer. So I use C13 carbon. What's the difference? We are all having abundant C12 carbon in our system, in all the molecules, but 1.1% is C13 carbon. It's a stable isotope, it's not radioactive, it's a very stable isotope, and on the planet, 1.1% of the carbon is C13 carbon. So we do have a low level of this stable isotope in our body. What's the difference between C12 and C13? You go back to school atomic physics, you'll find that in C13, you have one extra neutron in the nucleus, okay? You have the electrons in the nucleus, you have one extra electron, that's all. It's a stable atom, and you can use this to track metabolic flux. So what will I use? I can use labeled C13 glucose, where every carbon in glucose is C13, and I can follow this up by mass spectrometry. Mass spectrometry will differentiate between C12 carbon and C13 carbon because 1 Dalton is a fairly big number for modern day mass spectrometers to distinguish between a product which has got C12, a metabolite which is all C12, and maybe it has 1 or 2 C13 carbons. So I can follow that flux. I'll show you in the next slide how we do it. I take a petri dish where my cells are growing. They were originally growing with just ordinary glucose. I remove the ordinary glucose, which is C12 glucose, with very low percentage of C13, 1.1%, which is naturally abundant. I replace it with 100% C13 glucose. And now I have shown fumaric acid over here. If it is all C12, I'll have the mass that is expected. So I say M plus 0, nothing has been added to it. If there is one C13 that is added, it is mass of fumarate plus 1 Dalton, because one of the carbons is a C13 carbon. The difference is mass from C12 to C13 is 1 Dalton. Similarly, if I add 2 C13s, it will be mass plus 2. 3 C13s will be mass plus 3. 4 will be mass plus 4. You have to follow this because that's what is going to be the next bit of my talk. So what do we do in mass spectrometry? We take all the metabolites out of the cell, extract it out, put it through an HPLC system. They separate these molecules, and then those molecules that are getting separated go into the mass spec. The mass spec detects the mass by charge ratio of every single molecule that goes into the system. Yeah, so if fumaric acid is getting labeled, it will tell me how much of C12 is there, how much of 1 carbon, which is C13, is there, how much 2 carbons of C13 fumaric acid, 3 carbon fumaric acid, 4 C13 fumaric acid is there in the cell. That tells me what is the flux of metabolism within the cell itself. So these molecules of fumaric acid, which may have 1 or 2 C13s, They're called isotopologues. They're isotopes of the same molecule. They're isotopologues. Only thing is C12 has been changed by C13. I can follow the traffic that happens in the cell, the metabolic traffic that happens in the cell. Okay, so let me take you through a simple example. There is glucose. All the filled circles are C13. I have put two colors for C13 as shown over in the little legend over here. It can be red or black. C12 is an unfilled circle. So let's say we are now actually going through metabolism. So that glucose through glycolysis that you all know gets converted to phosphoenol pyruvate. If you recollect, phosphoenol pyruvate gets converted to pyruvate. Pyruvate can enter into the mitochondrion. So let's follow it. Yeah. So phosphoenol pyruvate will have 3 carbons, which are now C13, because that's the glucose that you're feeding to the cell. Pyruvate will have 3 carbons, which are C13, and lactate, which is produced, also will have 3 carbons. Pyruvate enters the mitochondrion. 2 carbons are now C13. Then it goes through the cycle, and you can have multiple rounds of the cycle, and you can have, this will be the pattern of labeling of each of these TCA cycle intermediates. Labeling how many C13s are there, what are the different isotopologues that metabolism produces. Yeah. Simultaneously, there are reactions happening outside the mitochondrion. Phosphoenol pyruvate can get converted to oxaloacetate, oxaloacetate into aspartate. Aspartate, as I showed you early on in the slide, feeds into AMP synthesis. And these are also C13. I put a different color only because to differentiate between the mitochondrial compartment and the cytosolic compartment. Yeah. That can enter the mitochondria because we know it enters the mitochondria. Initial NMR studies show that. It enters the mitochondrion, and I've just put different colors for the same C13, but they're trafficked through 2 different pathways. And this is the fate. And you can map all of them. So I've taken only the TCA cycle, but if I put glucose, it can go into pentose phosphate pathway, and the ribose ring will be also labeled. It can go many, many places. This is a simple picture for you to see how I can track metabolism by using a stable isotope food nutrient in the system. Instead of glucose, I could have used glutamine. That also gets in. We did that too. And this is how it would look. All these M is the natural mass. If C13 is added, I've written how many C13s could get added. And that's the flux that's happening. Flux, when I knock out a gene, flux can get completely impaired. I'll tell you, I won't go through every aspect of the flux. The TCA cycle flux was heavily impaired, especially the glucose-derived TCA cycle flux. But the glutamine flux, to some extent, was operating. But what we found interesting was inosine monophosphate. I told you parasite doesn't de novo synthesize purine nucleotides. It can only salvage the purines from the human host, convert it to IMP, which is the precursor for GMP and AMP. The +5 labeling in IMP was dramatically higher than wild type. What is +5? Why is IMP, which is inosine monophosphate, now having additional 5 daltons? That is the reaction that happens in the cell in the parasite. Hypoxanthine, which is a purine base, reacts with phosphoribosyl pyrophosphate, and there is the ribose ring over there. Since I have put in glucose, which is labeled, pentose phosphate pathway is operating, that ribose ring has got labeled, and that is the ribose ring which is getting added on to hypoxanthine to give rise to IMP. So my pentose phosphate pathway has somehow, the nucleotide synthesis has increased, pentose phosphate pathway flux has also increased when I knock out FH. Fumarate hydratase. Okay. What is produced in the pentose phosphate pathway? I have a cell in which much of my glutathione has been modified. It is succinated. I don't have the free thiol. There's enormous oxidative stress in the cell. What does pentose phosphate pathway, apart from producing ribose, which is labeled produce, it produces NADPH, which is a key reducing agent in the cellular system. when I knock out fumarate hydratase and reduce the TCA cycle flux, the parasite produces NADPH through the TCA cycle also. I've impaired that because I've knocked out one key enzyme in the TCA cycle. So I have oxidative stress coming from this unexpected succination of glutathione. I have oxidative stress coming from reduced levels of TCA cycle produced NADPH. The cell responds. How does the cell respond and try to survive? It tries to survive by increasing the flux through pentose phosphate pathway. It will produce more and more of NADPH, which is a key reducing agent during all conditions, including oxidative stress. Where does NADPH? It feeds into many things, but where does it feed in? One key thing in the context of this oxidative stress is the fact that it feeds in to the generation of reduced glutathione. Glutathione can be oxidized, glutathione can be reduced. The glutathione reductase converts oxidized glutathione, GSSG, to reduced glutathione, and you need an NADPH for it. So if you want to keep the oxidative stress under balance, this needs to be operating. For this to operate, I need NADPH. So the cell has rewired itself to increase flux through the pentose phosphate pathway, produce more NADPH levels. To summarize, what have I seen? I have seen an increase in fumaric acid level. The increase in fumaric acid levels now modifies many cysteines, including that of glutathione. The free glutathione that is available is reduced. The cell responds by increasing NADPH levels, which will help in converting oxidized glutathione to reduced glutathione. And during these asexual stages and gametocytes, it is always intracellular within the human erythrocyte. And the human erythrocyte is a good supplier of all these metabolites, including glutathione and NADPH. But when it goes and forms gametes, which fertilize and form zygote and ookinete, which is the pathway, which is the stages, developmental stages which are impaired, they're all free. They're not intracellular. They don't have access to these metabolites, which in the erythrocyte, there's all plenty of everything is there. So that is where we have complete developmental arrest, and there's no development beyond that. And this provides the metabolic basis of why there is a developmental arrest at these stages. Beyond the phenotype, you can now explore metabolism in these organisms. Do I have a similarity to pathways which are involved in glutathione synthesis itself in terms of the phenotypes that we see? Yes, indeed, we do have. Shown here in this bottom diagram are two knockouts that people have made. One is the glutathione reductase GR, which converts oxidized GSSG to GSH. If you block that, asexual stages are fine, but gametes, ookinetes don't develop. Mosquito stages are completely impaired. You are playing around with redox metabolism because you've knocked out glutathione reductase, which is GR. Similarly, if you knock out gamma glutamyl synthase, asexual stages are fine. They take everything from the host. But once you go to the mosquito stages where they are free living, this aspect of redox metabolism becomes very, very important. And Dr. Jyoti Prasunna wanted me to talk a little bit on, how can I identify a target and drug development. I'll just show you a little bit on it. Fumarate hydratase, what does it do? As I've been telling again and again, it converts fumarate to malate. It just adds water molecule. Double bond is gone, and a hydroxyl group is added. In Plasmodium falciparum, you have a fumarate hydratase, which is oxygen sensitive because it has an iron sulfur cluster. I think you would have heard of iron sulfur clusters, 2FE, 2S clusters, 4FE, 4S clusters. The Plasmodium enzyme has an iron sulfur cluster, that the picture shown over there. And that iron sulfur cluster we established is required for the catalytic activity of the enzyme. Two classes are there. Class 1 contains iron sulfur cluster and is very highly oxygen sensitive. Class 2 is no iron sulfur cluster and is insensitive to the presence of oxygen. What do parasites have? Parasites have this class 1, whereas the human host has the class 2. We looked at these structures, we solved one of them, and if you see, the left extreme is the human enzyme. It is a tetramer, it catalyzes the same reaction without the iron sulfur cluster, whereas if I take from Archaea, from Leishmania, the plasmodium is a model structure, the first 3 are crystal structures. If I take the crystal structures, you can see they have no similarity in structure at all, but they catalyze the same identical reaction. So you have the same chemistry being done in the human host by an enzyme whose structure is very different, and the way it catalyzes the mechanism is similar, but it doesn't require an iron sulfur cluster. On the other hand, the plasmodium enzyme and all parasitic protozoal enzymes have this different structure, and it has that iron sulfur cluster that is shown over there. So we exploited this iron sulfur cluster being present in the parasite enzyme. That's the product of the reaction. Malate, fumarate gets converted to malate, and that's shown on top over there. Malate has got hydroxyl group. So we ask the question, if I remove the hydroxyl group and put a thiol over there, I get mercaptosuccinic acid or thiomalate. Will thiomalate, instead of a hydroxyl group, inhibit the parasite enzyme? And what will it do to the human enzyme was the question. We found that it inhibits the parasite enzyme with nanomolar Ki values, so potent inhibitors in the nanomolar range, whereas even at 10 millimolar concentration, it does not touch the human enzyme. Thiomalate is non-toxic. It is used for many therapies. It's a conjugate that is used for many therapies, and thiomalate is actually clinically used as a conjugate. So the question that we are trying to address now is fumarate hydratase, an enzyme which can now be used as a transmission blocking drug. There is a great emphasis on developing what is called transmission blocking drugs. That is, you not only kill the parasites, you also tamper with the gametocytes so that you can be a carrier for a long period of time, gametocytes freely flowing in your bloodstream. To prevent transmission, it can be developed transmission blocking drugs, and thiomalate, in our opinion, could be a potential transmission blocking drug because it affects the gametocytes, gametocyte development into kinase, and this development, if it's affected, your transmission is going to be blocked. Finally, I want to tell you that metabolism is, people would think it's all studied, done, books are closed, and it's no point studying it because it's also difficult to understand metabolism. Shown here are very recent papers, and you are focusing on stem cells, very recent papers by which same mass spectrometry which I described to you can be utilized to look at fluxes which change upon different kinds of cell types, a malignant cell versus normal cell, a cell which is undergoing Warburg effect compared to a cell which is aerobic respiration, and various aspects now. For example, this is what I teach in my class under metabolism is, we think lactic acid is a waste. It's just thrown out. But going back from Cori cycle, you can see over here our understanding of lactate as a fulcrum in metabolism. And every aspect of this, to understand even microbial metabolism, the use is heavily dependent on mass spectrometry. There are large numbers who are using NMR2. It's less sensitive, but you don't need to separate out any molecule. You can use blood or plasma as it is to look at variation in metabolism across individuals with different control population with specific disease states, and both mass spectrometry. And now even NMR, the new techniques which have come up by which you can follow many of these metabolites across different states and individuals in cell types, everything. So it is indeed a major change that has happened in trying to understand metabolism back again in the context of how do fluxes change when cells are functioning in a very different manner. Before I close, I just took the statement from JBC, Journal of Biological Chemistry. I talked of anaplerosis of fumaric acid. Anaplerosis and cataplerosis work together to ensure the appropriate balance of carbon flow into and out of the TCA cycle. The beauty of this fundamental biological mechanism is undeniable in its simplicity, but ponderous in its complexity. I hope I have conveyed this to you, how, looking back at metabolism throws absolutely new light into the way we understand the whole process itself. Thank you for your attention. And these are the students who have worked towards understanding metabolism and the parasite. We have a good mass spectrometry facility which enabled me to carry out these studies, funding, and finally JNCASR, which has been a place for me to carry out my research. Thank you very much for your attention.
Thank you so much, ma'am, for the insightful talk. We will now have a short Q&A sessions. The audience is welcome to ask their questions.
Oh, great talk. I mean, the scary part about metabolism that we all students face, but at the same time seeing the actual effect of how metabolites play an important role, not just in humans, but also like a parasite was interesting. I have just one question regarding the inhibitor. So it was very interesting to see that the inhibitor is very specific for the Plasmodium, but what kind of an inhibitor is it? Is it binds to the active site, or is it like an allosteric inhibitor? Correct. Okay. Correct. Also, follow-up question. Will not affect. Correct. I see. Because if it is inhibiting the mechanism is through the iron sulfur cluster bindings, then there are a lot of enzymes within the system, like mitochondrial membrane proteins, that carry out TCA cycle, or not TCA, oxidative phosphorylation. They also have those iron clusters. So I was wondering whether that has any effect on the regular metabolism of oxidative phosphorylation, for example. Correct, correct. Right. Thank you.
Yeah, this is just technical question. One of the slide you tagged GFP with fumaric acid. Yeah, so it's a clone base. How? Because this enzyme you tag, not the metabolite. So okay, that's yeah. No, that I thought the metabolite of fumaric. No. That was a surprise. Okay, that's fine then. Yeah.
the cell, what does it do? It keeps on depending upon the, forcing the the below part to produce the enzyme, or it moves on to another metabolic pathway? Yeah. Why I'm asking is because this is the common way of getting resistant from, whatever we are giving from outside. So, can this be target potentially, or it's still in the dilemma too? Okay. Thank you.
So, not with the perspective of parasite, but you told that FH deficiency also causes cancer, renal cancer especially. So it is not the deficiency of the malate, but it is the accumulation of the fumarate. Am I right? Fumarate. So there is no any way of clearance of the fumarate. Is there any strategy involved in the clearance of the fumarate? Because majorly it goes for a post-translational modification, making it succinate. So you have epigenetic changes. HIF1 protein, yeah, fine. But cell is not thinking of clearing of the fumarate by alternative pathways. Yeah, yeah. Succinate. Okay. But in bacteria. Thank you. Okay, thank you.
If there are no further questions, we can move to the next part. May I now invite... Hello? Yes. Yeah. May I now invite Dr. Jyoti Prasanna to kindly come forward and present a token of appreciation to Dr. Hemalatha.
Keep this much.
A heartfelt thank you, ma'am, for sharing your wisdom and igniting our passion for science. A big round of applause once again, please. We will now take a short coffee break. I request everyone to kindly come back in the hall by 11:30 sharp as we can resume the program. Thank you.
We at MIRM, we offer two master's program, and this year we introduced another master's program. It is research intensive programs. We have MSc in stem cells and regenerative biology. We have MSc by research in translational neuroscience, and this year we introduced MSc by research in translational immunology.
Each of the class is associated with a practical session in the evening. We offer 3 programs here: Masters in Stem Cell Technology and Regenerative Biology, Masters by Research in Translational Neuroscience, and Masters by Research in Translational Immunology.
I'm so grateful and happy to be a part of this institute. I've always wanted to do a course on stem cell technology, and I know I've come to the right place. And as you have already seen, we have PhD labs where all the JRF and PhD students do their molecular bio work like PCR, Western blot, RNA isolation. And then downstairs we have the culture lab, we have tissue engineering lab where we do 3D bioprinting. Yeah, all the labs are very advanced, and I'm so happy to be a part of this college, and I would totally recommend it to the future aspiring scientists and researchers.
science isn't about just theories, it's about discovery. Every time I analyze a data set, I'm uncovering new ways to understand the brain and the complexities that shape who we are.
Studying immunology isn't just about learning how the body fights disease. It's about witnessing it in real time. Every experiment here brings me closer to understanding how we can strengthen our own defenses.
Stem cells aren't just the foundation of life. They are the future of medicine. Learning how to guide them towards healing is more than research. It's about transforming what's possible.
Mic check. Okay. Yeah. We're about to start the second session. I request everyone who is standing outside to please come in. Good morning, everyone. Welcome back. I hope the coffee has kicked in and the conversations might have sparked a few great ideas. Welcome to the second session of this National Science Day event, and thank you for attending this, and we are very grateful for all of the people who are present here. For those who may have joined us after the break, let me introduce myself. My name is Ilyama Punuskovur, and I will be hosting this second half of the session. Now let me start with all of the introduction. The very backbone of any scientific institute are indeed the professors. As for MIRM, I would say that the professors are the stem cell niche of this institute. Just like a stem cell niche provides the essential signals, development, factors essential for the proliferation, development, and the essential elements to develop it into its proper structure. Our professors are no less than that exact same intellectual microenvironment, thereby nourishing young minds. It is because of their contribution, their leadership, and mentorship that our institution stands as a center of excellence and academic advancement. Now I invite Professor Anujit Kumar, whose presence has always been a growing factor in this institute, to please come up to the stage and kindly present the faculty introduction.
Good morning. Happy National Science Day, along with the coming Women's Day also. As you know, yeah, science thrives, perseverance, innovation, curiosity. Along with that, probably the woman has played a very significant pivotal role in shaping the future research. Probably it has come to the stage where without women contribution, there is no science existing. It has gone to that stage. On this occasion where we are celebrating National Science Day, together with the Women's Day also, which is coming, and the topic which is there, the Science Day, Women in Science, Catalysing the Viksit Bharat, it's very apt actually when you consider with the perspective of V Raman actually, because the story goes that V Raman was not encouraging women to be in science. In the previous one, Professor Hemalatha Balaram was showing that Kamala Sohani was the first woman PhD holder in the country, and V Raman was the one who was opposing her PhD. Okay, so Kamala Sohani did the hunger strike in front of V Raman's house actually. It was in IISc. So that was, this is the stage where we are, as I told that, without women, there is no science existing. On this occasion, I take this immense privilege to introduce the co-faculty, Dr. Shagufta Parveen, who is the speaker of today. So Shagufta has an 25 years strong research experience, teaching experience. And she has been there from past 18 years in MIRM. She has been the integral part of MIRM, if you would say like that. And she's a very dedicated teacher, and she feels the enthusiasm in the young mind. And actually speaking, she's the spirit of MIRM when it comes, because she is the coordinator of stem cell course. okay? And she has got international publications, and also, if you look at her, she's an active participant in majority of the events what is going on in MIRM. And one thing, yes, when I looked at the CV, I saw that she is a rank holder in the university during her master program. She has cleared her CSIR UGC exam also. Then she had a teaching in other colleges, in the PS college, and then she joined MIRM. And since 18 years, she is contributing to the upliftment of MIRM. Okay? So, taking this small time, I would like to say that in the strong pursuit of the Science Day, the power of science and the immortal spirit of the women's, I would like to welcome Shagufta Parveen for this talk. Please give a big hand for Shagufta Parveen.
Thank you, Anujit, for introducing me. Good morning, everybody. Today I have prepared a talk which was called the self-organizing properties of, it was called self-organizing by pluripotency. It's there in the first slide, we can't see it. Anyhow, I would like to start by speaking a little bit about human development. Human development is very complex. We all start from a single cell called the zygote, which is a big cell. The zygote continues into dividing quickly into a structure or a ball of cells called the morula. The morula, if you can see the picture over here, the size of the zygote and the morula seem to be the same. That is because the zygote is a big cell and the morula is comprised of smaller cells. The morula quickly transitions into a blastula. The blastula is a fluid-filled structure, and it is made up of two types of cells, the inner cell mass cells and the trophectoderm. If you can see on top, I have labeled the days in which these transitions and developments happen. Between days 6 and 7, the blastula implants itself into the placenta or the uterine wall. And within another 2 days, the blastula is completely covered by the uterine wall. Then gastrulation starts. The structure, stage 7 is the gastrula, which is seen over there. The gastrula has the primitive string. You can see a small line that is there at the bottom of the picture. That primitive string elongates to form an elongated gastrula in an event called anterior-posterior elongation. Now, after this, there is the formation of the neural tube or the neural trunk, which consists of the nervous system and the somites, that is the mesodermal system. This transitions into a neural stage. Then in the neural stage, there is the formation of the neural tube, the formation of the somites, and there is the closing of the neural tube, which occurs. Then I've just skipped a few stages and presented and put up a picture of stage 14, in which there is more development. The head is formed. Below the head, you can see the cardiac regions, the liver regions which are forming, and you can see the appendages, that is the limb buds which are forming, both the anterior, the forelimbs and the hind limbs. And by day 60, we are more close to completing development, where we have a fully formed head. We have properly formed hind and forelimbs. Our digits are separated. They are no longer combined together. There are the eyes, the ears, the brain is forming. So what I want you all to pay attention to is that within 60 days, which is a short period of time, most of our organ systems are developed, and we, after day 60, it is more about growing big and taking shape. Now, why am I talking about human development? Because my lab is interested in understanding cellular and molecular events that are that govern human development. Now, during human development, the first few stages, that is up to the gastrullar stage, all of the stages are self-organizing. The embryo has the capacity to organize itself without much of maternal input. Most of these stages can be modeled in vitro. It is only after the gastrullar stage that the growing embryo requires input of the maternal input from the maternal uterine wall and its assistance and support. The early stages of human development is a self-organizing event. Also, I'm interested in understanding aberrant human development, what causes pregnancy failures, and and we are also studying some kind of congenital malformations. So my lab revolves around these kinds of studies. Generally, human fecundity is very low. That means the number of people being born is much less than the number of eggs that are getting fertilized. This is a pregnancy loss iceberg. So if we start from conception, which is at day 0, about 30% of the pregnancies fail within the first week of fertilization or conception. Within the next 7 days, another 30% of the pregnancies fail. So about 60% of the pregnancies fail within the first 15 days of conception. Now, if these pregnancies are failing, the mother would not notice it. These pregnancies fail without getting noticed. If the embryo is able to develop a little more and go beyond day 14, a month or 2 months of gestation, then, and after that there is miscarriage, then the mother will be able to notice those miscarriages. About 10% of pregnancies fail after day 14, and only about 30% of human beings are actually born complete. So 70% of fertilized eggs do not get formed, or they are not being born. What are the major causes for these pregnancy losses? There can be genetic causes. There could be some genetic abnormality or a chromosomal aberration that is there in the embryo, which is not allowing the embryo to develop completely. There can be poor uterine and endometrial health. There could be poor endocrine and metabolic health of the mother, the lifestyle of the mother, and the surrounding environment of the people. There could be the environmental factors like teratogens and infections. All of these could cause pregnancy losses. Now, to study human development and aberrant human development, we need a cellular system which has a developmental potency matching the growing embryo. The best possible system to work with is the human embryo itself. The problem with working with human embryos is that, A, they are very limited to access. We cannot get human embryos to work. There are some IVF embryos which are available, but it is very stringent to obtain those embryos. Another thing is that these embryos are quickly transitioning through development. So it is not, we are unable to hold them in a particular state for a very long time because they move so fast through development. We can't capture them in any one state to study that state effectively. What scientists have done is, within the embryo, there is the inner cell mass, which is the pluripotent cells, which becomes the embryo proper during development. Scientists have isolated these cells, and they were able to capture this pluripotent state in vitro. So this in vitro capture, it is a very strong tool for us to study development because those cells can mimic or recapitulate most of the events of development. Now the problem with these kind of experiments is that there is limited access to these embryos, and there are ethical concerns which are surrounding it. Every time we use an embryo for research, it is like killing a human being, and there will be it's not very well appreciated or encouraged in many countries. The other way of getting pluripotent cells is to reprogram somatic cells. We can take any somatic or an adult cell and reprogram them into pluripotent-like cells. So this is a very efficient method to generate pluripotent cells. Now, be it from whichever source, whether they are from the embryonic source or from the iPS technology, these cells, when you culture them with specific cues, they have the property of self-organizing. So if you put them together with proper cues, they can self-organize into various structures, which can be very useful for us to study human development. What we are doing in the lab is, like I said, we are interested in pregnancy failures and congenital abnormalities. We are obtaining fetuses which have which have either aborted spontaneously or which have some kind of malformation. We are isolating fibroblasts from them and reprogramming them. And of course, for every experiment, we require a control. So we have some control, we are generating some control cell lines also. Now, these pluripotent cells, the pluripotent cells, within the embryo, there are ranges of their pluripotency. Like, for example, the blastocyst, through implantation and complete covering by the uterine wall, the state of pluripotency is a range. There is a naive state, formative state, and a prime state. Naive state is more potent compared to the prime state. When we isolate these cells from the blastocyst and grow them in culture, they somehow acquire a prime state. Though we have isolated from a naive structure, they acquire a prime state because we are growing them in FGF and activin. Recently, in the past few years, there have been there are lots of small molecule cocktails which have been developed, which can push these cells back into a naive state. Most of these molecules, they are kinase inhibitors. So if you use a combination of kinase inhibitors, it pushes the prime state back into a naive state. The naive state of pluripotent cells in vitro, it has a pre-implantation identity. They have extra-embryonic differentiation potential, and they have a low DNA methylation. So the DNA methylation is low, so it's like a clean state, and we can write what we want. We can instruct the cells to develop into any one specific lineages. Whereas the prime cells, they have a post-implantation identity, and they are poised for gastrulation. That means they can differentiate into only mesoderm, ectoderm, and endoderm with high efficiency. They have very low extra-embryonic differentiation potential, and they have a high DNA methylation pattern on their genome. On the right side, I've put up a figure which says how these small molecules are used. If you look at the top of the table, if you apply LIF and activin, the cells seem to be pushed more towards a naive and formative state of pluripotency, whereas if you use FGF, they seem to be pushed towards a prime state of pluripotency. Then there are some kinases like Wnt activators and MEK inhibitors, which if you apply to these cells, they get pushed towards a naive state of pluripotency. There are lots of these cocktails which have been developed, which is written on the lower side of the table. Now, chromosomal aneuploidies or chromosomal instabilities are the major cause for pregnancy failures or abortions or malformations. We call them embryonic lethalities. There are lots of reports which suggest that any kind of chromosomal abnormality leads to abortion or leads to embryonic lethality. And these chromosomal aneuploidies, they usually arise in early stages of development. That means during the blastomere stage and the morula stage. These kinds of abnormalities also come into existence when we are culturing these cells in vitro. So if you take these cells and grow them in vitro, they also tend to accumulate these chromosomal abnormalities. So what is it about these cells that supports or encourages the chromosomes to become aneuploid? Or there must be something that is happening that is not allowing proper segregation of chromosomes to occur in these cells. What I've shown over here is chromosomes just after, during the mitosis phase, the chromosomes, they get segregated into the towards the two poles. by the microtubule apparatus. The microtubules attach to a specific molecular structure on the centromere called the kinetochore, which is which is many molecules. It attaches to the kinetochore, and the two chromosomes segregate apart into two daughter cells. Now, if one of the chromosome doesn't get attached to a microtubule assembly, like if you see the one on the right is not attached to the microtubule assembly. I've also put a picture over here. If you can see where the asterisk is, there is one chromosome which is lagging. That means it has not gotten attached to the microtubules, and hence it is unable to get segregated into any which pole of during cell division. Now, when this happens, there is a set of proteins in the kinetochores that are called the spindle assembly checkpoint proteins. These proteins, after events of phosphorylation and recruitment, what they do is they form a complex called the mitotic checkpoint complex. And once this complex is formed, it delays the exit of, it delays the cells into progressing into the next phase of the cell cycle. So the cell cycle is held in that phase till the erroneous attachments are corrected. Then the chromosomes can separate accurately and cell division occurs. If any of these molecules is inhibited, or if it is mutated, or there is some other reason for them not functioning properly, what happens is the mitotic checkpoint complex does not form. If it is not forming, it allows the chromosome missegregation to occur. We can experimentally recapitulate this by using a molecule called reversine, which inhibits the MPS1, the MPS1 component of the MCC. Now, when we do that, what we found is that we could, when we inhibited this complex, we could introduce a lot of chromosomal anomalies into the cell. If you see the karyotype of these cells, it is nearly tetraploid, except for 3 or 4 chromosomes, which are 3 in number. The cells could really accumulate a large number of chromosomes within them. Now, these cells were surviving. There was nothing going wrong with them. They were surviving, except that they were larger in size compared to normal pluripotent cells. So we have shown that when these cells have such kind of chromosomal missegregation events, there are some differences in cell cycle proteins and increase in apoptosis. And also we showed that because of the increase in the number of chromosomes, there was a difference in the dosage of genes and hence the dosage of the proteins, and the cells were really looking very big. Tressa is working on this project, and what she is trying to understand is what is going wrong during early stages of development. She is using a variety of cells, starting from fibroblasts, prime cells, and naive cells. So she has a range of potency, starting from low potency naive cells to, high potency naive cells to completely specified cells, fibroblasts. And she's looking into how these checkpoint proteins are behaving in them. She's just started off the project, and she's still looking at the transcriptome expression of these proteins. Now, like I told you, we are interested in congenital malformations. One of the malformations that we work with, or the abnormality that we work with, is Turner syndrome. Turner syndrome is a 45X monosomy. That means one X or Y chromosome is missing. About 99% of embryos with this kind of monosomy abort. That means they don't make it to full-term birth. Either they abort spontaneously, either they develop to a certain extent and then abort, or it is an unnoticed abortion. This is a picture of a first trimester abortus fetus. Now, what we have done is we've taken one fetus like this, converted the cells into iPS cells, and we are understanding what is going wrong during early stages of development of Turner syndrome. This work is from James Turner's lab. What they have done is they've taken, from mouse, they have generated 45XO, they've generated X monosomic blastocysts, and they are looking at how the distribution of cells is there in these blastocysts. So in the blastocysts, there are two lineages of cells, the trophectoderm and the in a cell mass. They are looking at how the cells are distributed in these blastocysts. So if you look at the exo, the blastocyst is very small, and then the way the cells have been specified is very different. The gray marks uncommitted cells, the green is ICM cells, and the blue is the trophectoderm cells. If you look at the distribution of the cells in the exo embryo, it is only about, it is uncommitted or trophectoderm lineage. We wanted to do similar kinds of experiments with the Turner syndrome embryos, but it is impossible to get Turner syndrome embryos because A, we will not know that it is a Turner syndrome, embryo, and it is not possible to get 45 exo embryos. So what we did in the year 2021, there were a series of papers which showed how to convert pluripotent stem cells into structures called blastoids. So these blastoids, they are very similar to human embryos, and it's like a continuously evolving field. There are new ways of generating these blastoids. We wanted to see if we could make Turner syndrome embryos or 45 exomonosomic embryos using these technologies, and yes, we could do it. We have generated blastoids from 45 exo cells. We have also generated blastoids from 46, that is 45 with 46 with a Y chromosome. We are yet to generate blastoids from the female lines. Now what we want, now the problem with generating these blastoids is to manage their size. Each embryo, each blastoid would have different number of cells, and that in turn would distort the distribution of cells into the lineages. However, there are finer methods of cultivating these structures. We can really count the cells, and there are these microwells which allow us to more or less regulate the cell number and come about with structures which are relatively close to natural embryos. This work was done by Anjan, who was one of the master's students in my lab. Now, moving ahead in development, what happens is we will there is a formation of the neural tube and the trunk or the axial elongation somites and other structures. We are also very interested in understanding how neural tube defects are formed in the growing embryo. It is a serious birth defect. It is one of the most common congenital malformations. It happens because there is a failure of the neural plate to fold into a proper neural tube. There are several forms of these neural tube defects. They can either be anterior, where there is improper closure of the head region, or they could be posterior, where there is improper closure in the lower back. In India, for every 1,000 births, about 4 births come up with these neural tube defects. There are various factors which govern these defects. It could be genetic, but what seems to be the most talked about and the most common factor is maternal folate deficiency. So how this folate is getting metabolized by the mother seems to play an important role in neural tube defects. The planar cell polarity program also is implicated, along with other pathways which have been implicated. What we are interested in is in understanding how folate deficiencies cause these neural tube defects. Using pluripotent cells, we can come up with structures called neural cysts, which is shown on the blue structure over there. It is very similar to the closed neural tube. The identity of the cells is also very similar to the closed neural tube. So we can use these structures. They are very simple structures. If you grow them in simple media and put them in an ECM, we can form these structures. We can use them to understand some basic aspects regarding closure of the tube and how we can and apply different molecules to it to understand what is happening in neural tube closure. Like I said before, there is a strong association between folate deficiency and neural tube defects. Tejashri from my lab is taking a look at these defects and trying to understand what is happening. Now, within our cells, we have the folate cycle, which metabolizes folic acid. The folate cycle is it is working in synchrony with the methylation cycle. The methylation cycle, or the one-carbon metabolism cycle, it generates methyl groups. Initially, we thought that it is these methyl groups which are changing the epigenome of the embryo and hence causing neural tube defects. But now it seems to be that's not how it's working. The folate the folate cycle is also responsible for generation of nucleotides. What we are seeing is low proliferative rates, so we assume that the nucleotide pool is getting lowered because of folate deficiency and causing proliferative disorders. However, we are going to still take a deeper look into what is happening. So neural tube defects can also happen because of some teratogenic effects of drugs. Valproate is one such drug. It is an anti-epileptic drug. It is used to treat bipolar disorders. It can be used as a mood stabilizer. So it is a commonly used drug for various psychiatry disorders. It is a histone deacetylase, and there are multiple reports which have suggested that there is a strong association between valproate and neural tube defects. So mothers who have been taking valproate tend to give birth to children with neural tube defects. We use these simple cysts to try to understand what is happening with this kind of development. So these are cysts. We have characterized them for their properties. They express SOX2 and nestin, which are markers for neural progenitors. They also form this, because it's a closed structure, we wanted to check whether they were able to form a closed tube-like structure. So we checked for markers of apicobasal polarity like esrin and ZO1. They express more neural markers like beta-3 tubulin and N-cadrin. And they have very little bit of E-cadrin. There is E-cadrin, a little bit of it. And they express portocalcin, which is again a sialoprotein which is found in the lumen. When we apply valproate to these neural cysts, you can see that there is a complete deformation of these cysts. They tend to get smaller in size. They are not rounded anymore, and they are they have acquired some different shapes. The important thing is that the valproate is killing these cells, and it is not allowing proper apicobasal polarity to get established. When this is some of the quantification that we did on the cysts. So the size of the cysts goes down, the number of cysts goes down, and also the lumen diameter, it is variable. It is neither going down, it's not going down, but it is variable because there is a deformity in the types of these cysts which are formed. Similar kinds of experiments were done with spinal cord organoids. Again, we saw that the size of the organoids goes down with application of valproate on the spinal cord organoids. What was one important observation that we could get was, if you take normal cysts, and if you look at the ZO1 expression, ZO1 is a junctional protein which is found on the apical region of the cells. The more the constriction, we can look at the level of constriction looking at the ZO1 expression. So if you can see over there, I've just drawn one ZO1 or one constriction around a cell. It is small, it is quite close. Whereas if you take a look at the VPA-treated cysts, the ZO1 is broadened, so it is not able to constrict the cell enough. It is much bigger compared to the control cysts. So if we looked at some of the area of the ZO1 distribution, it seems to be very high in VPA-treated cysts. So if there are normal cysts and if there is constriction, and these were to make the if these are neuroepithelial cells, and if we are recapitulating neural tube bending and folding, there will be neuroepithelial bending because of the constriction of these apical regions, and there will be neural tube closure. Whereas if we are applying VPA, the apical region seems to be wider. There is insufficient neuroepithelial bending, and could be one of the possible mechanisms by which there is an aberrant neural tube closure. Then regarding more, Tejashree will be talking more regarding how she is trying to understand what is happening with folate deficiency and what she is doing in her experiments. Now, as we move along the development of the human embryo, earlier I told you that there is the formation of the primitive streak, the anterior-posterior axis is established, the embryo elongates in an axially, and there is generation of these somites and other structures which are formed, giving the embryo anterior-posterior, right-left, and a dorsal-ventral axis. Again, there are lots of, we can model these states also using pluripotent stem cells in vitro, that is somitogenesis, trunk formation, elongation of the anterior-posterior axis. All of this can be done using these structures. There are lots of papers, especially by Naomi Morris, who has shown that, for the first time showed that this kind of elongation was possible, and she called these structures gastroloids. And recently she has also published a paper where she's generated neural trunk. Neural trunk is when you have the neural tube and the somites generating, co-developing together to mimic something which is close to human development. We are also looking at how to generate these structures. Gastroloids, we are interested because I want to see what is happening in the Turner syndrome model. How is the gastrulation proceeding in the Turner syndrome cell? And we are doing the neural trunk to see how these structures are behaving when we remove folic acid or we add a teratogen to these structures. These gastroloids, they show typical axial patterning, anterior-posterior patterning. On the posterior side, there are these cells called neuromesodermal progenitor cells, which bring about the elongation of the growing embryo. These cells are positive both for SOX2 and brachyury, as you can see over there. And we have really not modeled this with, we are halfway through modeling this with Turner syndrome cells, but I'm really curious to see how these structures form, how they are patterned, and what is the type of cell distribution in Turner syndrome gastroloids. The neural trunk structures. Now, if you look at this, this is the Carnegie stage 11 of the growing embryo. You can see the head regions on top, the neural tube, which is on your right, and then somites, which are next to it. When we generate neural trunk structures, they are very similar to this stage of development. If you can see the red structure, the red staining on this neural trunk, it is very similar to the neural tube that is developed on this Carnegie stage embryo. They have a lumen within them, and they're developing. Next to it is the blue structures. The blue structures are the somites. I'm not able to get a proper feel, so I'm showing it like this. You can see the somites which are developing very close to the neural tube. So more or less, we can mimic a structure which is close to the neural trunk during the development. Moving ahead in stages of development, it is during stage 13 and 14 when the heart is getting formed. The formation of the heart, it is a complex series of events which involve bending, looping, forming of the four-chambered heart, and the valves that are formed in this heart. The valves, they prevent the blood flow from going haywire, and they regulate the directions in which blood is flowing through the heart. If something were to go wrong with valve formation, there would be mixing up of blood in the chambers and causing issues with the way the circulatory system is working. So the valves, they form at about 30 to 40 days during development. You can't see the picture. It is, when the heart is forming, there is a proper heart tissue which has a myocardium on the outside and an endocardium on the inside of the wall. Between this, there is an ECM called the cardiac jelly. Some cells from the endocardium, they migrate into this cardiac jelly, populate it, and these are the structures which form the heart. There are some neural crest cells also which migrate into it to form the heart valves. Now, in Turner syndrome, there are lots of abnormalities. Some of that 1% of embryos, they live and they form adult individuals. These adult individuals are females because they have only one X chromosome. These females, they have a lot of abnormalities in their phenotype. They have a short stature, they are infertile, they have a lot of autoimmune diseases, cognitive problems, and they have lots of congenital heart malformations. One of the heart malformations that we are interested in is the bicuspid aortic valve, which is found in most of these people with Turner syndrome. So we want to know how the 45X karyotype affects this bicuspid aortic valve morphogenesis. What we've done to do this is the latest stage of development, and we really at this, at that point when I started this project, I couldn't make a heart structure. There are some studies which are saying that we can have a heart organoid now. What we did was we decided to just take pluripotent cells and convert them into valvular cells. So if we give a series of cues to them, they can convert from a pluripotent cell into valvular cells. So we take iPS cells, we can make them into mesodermal progenitors, convert them into endothelial progenitors, endothelial cells, and then we can model the movement of these cells into ECM, something that happens like movement of the endocardial cells into the cardiac jelly. We wanted to mimic that in vitro. So this is a cellular platform. It's not a self-organized or a 3D platform. We take these cells and we gradually stage-wise differentiate them into endothelial cells, and then we just put these endothelial cells over a matrigel, which is an ECM, and we see how they are migrating into it and whether they are able to recapitulate the formation of the cardiac valves. So far, we know that when we put Turner's, if you can see on the right-hand side, the normal cells with a normal karyotype, they tend to migrate very well into the matrigel, and they form good mesenchymal cells, which we can see in the morphology, whereas the Turner syndrome cells, they have a little bit of inhibition in migrating into these matrigels, and they are more rounded and they are unable to acquire a mesenchymal phenotype, which is typical of a valve leaflet. Vaishnavi was another student who did this work. Now, because we are talking about cardiac generation, we can also do vasculogenesis with these pluripotent cells. So if you take iPS cells and give them gradual cues, we can make them into mesoderm, convert them into a vascular lineage, and allow for sprouting to happen, which you can see in the pictures over here. It's a day-wise. These cells, they start producing new blood vessel-like structures, and they start forming a network, which is quite elaborate. We are using this to understand how the secretome of placental mesenchymal cells behaves. So we are adding the secretome onto these structures and seeing whether they are able to sprout more, sprout less, remodel better, form better nodes, junctions, etc. Rachna and Yogesh. Rachna is primarily doing this project, and Yogesh also assisted in doing. Yogesh was a former MSc student in the lab. Okay, now finally, if you want a pregnancy to be successful, we require a very healthy mother. I'm meaning to say the uterus and the endometrium has to be healthy. There is a disease of the endometrium called endometriosis, where some of this endometrium tissue, it starts growing outside of the endometrium. It forms lesions across the uterus, ovaries, and other organs in the body. Mahalakshmi is studying this abnormality. She collects clinical samples of endometriosis, and she is, as of now, understanding the transcriptome signatures of it. What she aims at doing is do a high-throughput transcriptomic study, find out some drugs in silico, and apply them to these generate endometrial organoids, apply them to it, and understand if it is working in treating this endometriotic disease. Okay, so that's about it. So I just gave an overview of the different projects that we are doing and how it is related to the development of the human. First of all, I would like to thank the Manipal Academy of Higher Education for supporting this kind of research, and MIRM, which supports this kind of work to happen without any problems. I'm funded by a DBT project and a DRDO project with which I'm able to do these experiments. Thank you. All for me. The boys. If you have any questions.
Now we'll have a short question and answer session. Is there anyone who wants to ask Dr. Shagufta ma'am any questions related with the presentation?
Antidepressants. So will that affect, or is it only specific to this drug?
It's specific to valproate. Valproate has an association with neurodepressive, not other psychological.
Not other psychological. So they have to be, they have to wean the treatment?
If the mother wants to conceive, they wean off the treatment. It is difficult if it is an epileptic patient, but they do that.
Okay. Anybody else? Yeah, this related to your valproic acid project. So actually mechanistically how this working? Because valproic acid can stop when it's a heterochromatin state and stop division and proliferation.
We haven't looked at how it is changing the chromatin structure. We haven't gone that.
Sometimes it can make a quiescent state and completely stop. And in that state, if
we have to take a look at how the the acetylation patterns are there on the histones and on the. I haven't taken a look at it as of now. I haven't yet.
Okay.
Yeah, hi, nice talk. So the first part where you were talking about the spindle assembly checkpoint, you showed some of your data related to that. Maybe I missed it. Did you look at one specific member of the spindle assembly checkpoint?
So we have inhibited only MPS1, which is the primary phosphoryling kinase.
Okay.
So it's the phosphorylation by that kinase that actually triggers the whole pathway to get activated. We inhibited that. We haven't looked at anyone. As of now, we haven't looked at
the other. Okay, all right, thanks.
So post inhibiting the MPS1, have you looked at the differentiation ability of the cells?
Only up till the embryonic bodies, it does form cells of 3 down lineages.
Okay. For me, more interesting is because most of them form tetraploids. Am I right?
Most of them are forming tetraploids. We have to go beyond the embryonic body stage to see if they are actually surviving.
No, I would like to ask you, do they form extraembryonic tissues better?
I didn't check.
Because usually when there is tetraploid, they tend to form, they call tetraploid complementation you would have seen. So they tend to form more placenta-like structure than going ahead. So would they be
We didn't look at the extraembryonic lineages. We just stopped at the AB formation, saying that it goes through up till the gastrulation state. It is beyond gastrulation where all the other manifestations begin. We haven't started. We haven't taken a look at that. After we make them?
Yeah. So after that, can we use any CRISPR-Cas or some technology which can...
We can use them on the cells that we are making them with and then use them to make the structure.
So like any syndrome, if it is present, can you reverse it? Like
we could correct it in the single cell stage, and then these structures form within 2 or 3 days. It's only 72 hours when you can see the structure. It's not difficult. We just have to make an aggregate of it. So maybe if we could do the correction in a single cell state and then make these structures, we would get the answer.
Thank you.
How do you? Yes, we can't create the morphogenetic gradient as it is in the embryo, which is tightly regulated, and it is because the extraembryonic structures are giving something to the epiblast and the epiblast outside. It's from all sides, but yet the anterior posterior elongation seems to be a very strong mechanism. Even if you're giving the signals from also, like if the structure is there in a well-containing media which has signals coming from all the sides, it is still able to elongate anterior posterior. It seems to be. No, I am going to look at the Hawks chains.
Shagutha, one question. It is really amazing to see the developmental structure stages on the dish and how you are preparing them. I am really intrigued to understand what are the challenges that would be come across to translate these to the real, addressing the problems that you are starting with, the diseases, the defects in the reproductive system. How do you think you can overcome that and offer them?
I think we can use mouse embryos to study early embryonic development, but a mouse development would still give rise to a mouse and not a human being. So there are genetic differences which are there between mouse and human embryonic development. These structures seems to be closest that we can get to human development. They are not the exact events that are happening during development, but we are getting the closest to them. In my opinion, we can use them to study the abnormality and nothing more. There is a lot of debate that is going on regarding what we can do with these blastoid structures, whether we should continue growing. We can even mimic implantation. So should we treat the blastoids like blastocysts, stop their study after day 14, and what we are going to do? So blastoids seem to be more something that we can study full human development. But of course, it comes with ethical restraints and how we are using these structures. Generally, we have to stop all embryonic research at day 14. There is a day 14 rule where we stop these structures from growing any further in the dish.
Okay.
In the blastoid, did you look at the trophoectodermal population, CDX2?
I have a picture for it, but I didn't put the trophoectoderm. I've just given an overview of what we are basically doing with all of these structures and how we are using them for studying various things. Yes, we will be looking at the way these different lineages are distributing in the blastoid, how the trophoectoderm is, what its transcription signatures are, and how the ICM is. We will be looking at in detail into all of these aspects.
Seniors, having valve issues?
No, what we are doing is we are studying the bicuspid aortic. Normally, the aortic valve is tricuspid. In Turner syndrome, instead of forming a with 3 leaflets, it forms a bicuspid valve. So, what is going wrong in that valve development is what we are looking at. We are not.
It couldn't possibly be extended to the valve issues.
It couldn't be. At least we are not looking into it. We are not working in that direction.
I was just looking at the molecular players. Will there be similarities or differences in and how that can be extended to the conditions?
You can take normal cells which are generated like this and put them on some kind of biomaterial perhaps to make a flap-like structures. There are many groups which are working on generating these valves. We are not working in that direction. Govind is asking something. Govind is asking something.
Ma'am, just quick question. Regarding the endothelial cells, whatever you have mentioned. Ma'am, is it the problem is with the endothelial precursor cells or the differentiated cells? Because it's just the malformation of the heart actually. And it's the malformation of the head or the congenital defects, right?
Yeah.
So whatever you are saying is just because of the EPCs or the late completely differentiated endothelial cells.
In this experiment, what I showed, we looked at the completely differentiated endothelial cells and their endothelial to mesenchymal transition, whether they were able to do that properly is what the data that I showed. On the X chromosome, there are genes of TIMPs, which seem to be playing a part in this kind of abnormal development of these cells. So we are taking a look at that.
Ma'am, again, just to clarify it, so
And in these patients, you are not seeing any vascular defects apart from this valve deformation, right?
No, they have other defects also. There is like narrowing, aortic dilation, coronary artery disease, aortic dissection where they tend to break, the arteries tend to break apart. These are other kinds of disease, other kinds of malformations which are there. We are only understanding bicuspid aortic.
Thank you.
Okay. Thank you.
Thank you, ma'am, for such a thought-provoking presentation. And now I request the Dean, Professor Jyoti Prasanna, to present a memento to Dr. Shagufta Parveen. They say great ideas can be expressed in a few powerful minutes, and that is the spirit of Flash Talks. Now we move on to the next exciting segment where our talented MIRM scholars will present their research ideas in a very precise and dynamic manner. We have a row of 6 students, and now I call upon Mr. Alan Brito for the first presentation.
Okay, a very good afternoon to one and all. My name is Alan Brito. I'm a PhD scholar in Dr. Uttara's lab. And the topic for my presentation is understanding mechanism of salmonella infection from a stem cell perspective. Okay, sorry for the glitch. Okay, can we go on? Okay, now to begin with, let's look at the human microgut, okay, which possesses millions of defined complex microbial community. Okay, these microbes contribute to carbohydrate digestion. It's not happening, guys. I'm sorry. Let's see. Yeah. Allah Ta'ala. Pointer? Okay, let's see. Okay. Now this gut possesses millions of microbes which form the community which impacts in carbohydrate digestion, regulation of immune system, and defense against pathogens. Okay. Yes, give me a minute. Something is wrong with my screen here. I can't see things here. Fine. My screen usually gave up. Okay. Now, these gut axis contributes to a lot of functions like the gut liver axis, the gut reproductive axis, etc. Now, a disruption, or here in this term is called a dysbiosis in this axis, could lead to a lot of disease conditions like the cardiovascular diseases, immune dysregulation, etc. And what could be the possible contributing factors to that? Looking at it, we look at two intrinsic factors like genetics, metabolic diseases, and in case of eccentric factors, it is infections and pathogens and long-term antibiotics or therapeutics. Now, this is where my area of interest lies. A quick review of literature where we've all heard about Salmonella typhi, the typhoidal strain, but let me introduce you to Salmonella typhimurium, which comes under the class of non-typhoidal strains. If no one's heard it, typhimurium infects both humans as well as mouse models. And the prevalence in India has been quite concerning, and the fatality rate is around 14.9%, and these strains are gaining multi-drug resistance. Okay. And our lab is interested in understanding Salmonella. Now, let's begin with how this enters the host system. The pathogen enters via the oral route, via contaminated food and water. It passes through the gastric pH of the stomach, and it survives through it by activating acid tolerance responses. Passing on from the stomach, it goes to the intestine, surpassing the bile salts, and reaches the intestine. And upon reaching the intestine, it triggers inflammation. And what does inflammation cause? There's a disbalance in the microbiota population as well as the Salmonella population, where the Salmonella increases, and there's a balance, disbalance. And let's look at how Salmonella enters the host cell. It preliminarily uses two mechanisms, the zipper mechanism, or which is called the receptor-mediated entry, and the trigger mechanism. Trigger mechanism is a forceful entry. Salmonella uses these syringe-like structures or specialized systems called T3SS, where it secretes effectors into the host cell, thus forcefully entering, or it makes the host cell engulf into itself. Okay? And in the microbiota. So, now the response towards a microbial or a pathogen infection is these microbiota as well as the host cells produces ROS and other byproducts. And upon production of ROS, now to protect the microbe or the microbiota, the pathogens or the species in it produces hydrogen sulfide. Okay? Now, this is to protect itself from the cross activity or damaging its own DNA. But the Salmonella very cleverly takes advantage of this and uses these byproducts as an additional electron acceptor or an alternative electron acceptor, which helps in proliferation of the Salmonella. Anything which is given against the Salmonella is right now in benefit of it. Okay, thus it takes over the system. A small introduction to stem cells. Now, over the past decade, while analyzing lung biopsy samples of MTB patients or Mycobacterium tuberculosis patients, it was observed that MSCs had migrated to these granulomas and had internalized MTB, which was quite interesting because in case we've never heard about MSCs internalizing pathogens, but that's the very first cue that was followed. And followed that, Carnetal's group in 2017 went on to understand this interaction. They've used bone marrow MSCs and umbilical cord MSCs, and it was observed that these MSCs could internalize GFP tagged Mycobacterium tuberculosis. Followed on, they went on to study the proliferation of the pathogen, that's in that graph B, within the host cell, the viability of the host cell, and they went on to knock down using siRNA, they knocked down Becklin, which increased the internalized bacterial population. Further studies were done to check whether rapamycin or autophagy activated via rapamycin could bring down the population, and yes, rapamycin induction of these infected cells had brought down the bacterial population. Now this brings on, and yeah, finally, few reports have recently come out where it is mentioned that MSCs could help, MSCs in the body could help fight salmonella, our area of expertise. Now we are trying to establish a model, similar model with mesenchymal stem cells, that is DPSCs. Our lab primarily works on DPSCs, and we are trying to understand the interactions between DPSCs and salmonella. In the first panel, we have first the characterization of MSCs, because while isolating MSCs, we have to first characterize them. These are all primary cells. Okay. Followed on the strategy that I used in the lab. We culture DPSCs in this corner. If people are not visible, shift. We infect DPSCs with Salmonella typhimurium for an hour. Okay. Post 1 hour, we remove any external pathogens. Okay. But to address this further, we use gentamicin. So gentamicin treatment of these in the media clears out any extracellular pathogens. And post infection, we go on for different time points. Okay. 0 hour to 24 hours to understand what has happened. So the graph that you see shows CFU, or colony forming unit of internalized pathogen, where we've infected DPSCs with 3 different MOIs. MOI stands for multiplicity of infection or ratio of hosts to pathogen. So we've challenged these DPSCs with a ratio of 1:1 and 10 pathogens is to 1 and 100 pathogens, where in all the 3 MOIs there is internalization and there is replication of bacterial pathogens. And not able to see, sorry. There's a video of where it shows it's confocal of the 3D volumetric view, where it confirms there is internalization of salmonella. Okay. Moving on, we wanted to understand the fate of host cells as well as the internalized pathogens. Here you see a panel of 6 images which are DPSCs stained with actin 555 and GFP tagged salmonella and DAPI, where across different time points we see the bacterial population increasing, and around 18 hours we see the cells are circling up. Now we are yet to study that. Probably it could be cell death, anything. But to understand this further, is there a loss in viability? There is a graph where we notice that again DPSCs are challenged with 3 different MOIs. The viability of the host cell, the proliferation of DPSCs have slowed down, but drastically not decreased. So the cells are still viable, but the proliferation has been slowed down. This is in comparison with media. Okay, DPSC is growing in 10% media. Now we've asked the question. We wanted to quantify the internalization percentage within how many DPSCs can actually take up the pathogen. And to answer this question, we've used imaging flow cytometry, where it's microscopy and conventional flow combined together. So it's the same dot plots, but with the power of imaging. Okay? And we've stained these cells with CD44 V450 and GFP and Alexa flow 640. Now, how do we differentiate between internalized pathogen and external pathogen? Okay. We've skipped, so conventionally while staining with antibodies, there is a permeabilization step, which allows the antibodies to enter within the cell. We've skipped the permeabilization step. Okay. So when we add anti-cell Alexa flow 647, these antibodies very specifically bind to only the pathogens which are outside, because we are not allowing it inside. So this is the strategy that we use. So the pathogens are JFP tagged, okay, but when bound to the antibody, these are expressing in the far red range. So you will see an expression of red and green as well. So you will see the DPSCs which are stained with B450, then DPSCs. So this is how the image looks. That is DPSCs alone. These are DPSCs with bound bacteria. They are not internalized. So they have the Alexa flow 647 as well as GFP. That's why you see a red yellow expression. Okay, and these are DPSCs with internalized. So there is no bound bacteria but internalized, and we have DPSCs with both internalized and external. So to quantify, we went on, and while calculating, around only 0.8% of the MSCs had internalized these cells, and after 3 hours it is increased to 1%. Okay. Now we wanted to understand the transcriptome level changes in the host cell. Now to begin with, we wanted to know whether is it a receptor mediated entry. So we had a panel of receptors where we've showed a couple of them here, where we see TLR3 and TLR2 expression is not upregulated at 0 or at the point of infection. But dectin-1, now dectin-1 is, there have been reports where Salmonella uses dectin-1 to enter within the host cell. So we see an upregulation of dectin-1. Moving on further, post-internalization, once it's inside the host cell, what happens? Is it engulfed or is there an endosomal pathway activated? Yes, there was. We see RAP5 increase. RAP5 is an early endosomal marker. No changes in the RAP7 expression, and LAMP1 and LAMP2 have been upregulated, indicating, or we infer that the endosomal pathway being activated transcriptionally. We are yet to assess this at a protein level. Okay. Moving on to further, we wanted to assess the autophagy pathway activation. To begin with, we started assessing the NF-kappa beta levels. So upon inflammation, the levels of NF-kappa beta is activated or increased, and this indirectly activates mTOR, ALK1 or ULK1, ATG13 and SQSTM. Okay. Significantly, only ATG13 has been upregulated, rest all have been downregulated. Now moving on to the phagophore formation, the autophagosome and autolysosome, etc. So far we have the autophagy markers or the autophagy genes, ATG16, ATG8 have been upregulated. It has to be further looked into it. Okay. Now the future perspective is we want to try to assess the immunomodulatory properties of DPSCs, and we want to stimulate these DPSCs with autophagy molecules like rapamycin, and to check whether there is clearance of bacteria. And finally, to analyze the impact of bacterial infection on the differentiation potential of DPSCs. And thus, I would like to thank Manipal, MAHE, MIRM for the support they've given and for the work that we do. I like to thank SERB for the funding, and I would like to thank JNCASR, especially Dr. Ravi Manjitia's lab, who's been our collaborator, and all the strains that have been given or seen here are a gift from his lab. I'd like to thank Dr. Dipankar Nadi for his thoughtful comments and helping me to streamline my work. And finally, SciTech for the technical support they've given. I'd like to thank my guide and all my colleagues. Thank you. I'm open for questions. Yeah. Got it. Okay, thank you. Now, on a long term, so when I had mentioned there are two parts of interest or concerns. One is the infection, one is the antibiotic treatments which are given for gut infections. On a long term, they might be invasive and might bring about a lot of multi-drug resistance. Then that brings about our question or my final end of the goal, like where can we use these DPSCs to treat gut bacterial infections? So that is the connect that I have of my study, which has not been mentioned here. But on a longer term, can we give these for therapeutics? So in that case, where it is a non-conventional way of isolating DPSCs from the dental pulp, but can this be given for microbial treatment? So that's the connect. Mm-hmm. So ma'am, that model has still been understood in the lab. It's still under process of establishing. So even though there are papers and protocols with respect to monocytes or THP1 differentiated macrophages, they're still under work, has been to be looked at.
So my question is coming from the fact with which you have started that mycobacterial tuberculosis can sort of house in your mesenchymal stem cells and escape immune surveillance mechanisms. So the way the pathogen is processed is different in macrophages versus these stem cells. So what could be the mechanisms could be an interesting thing to check.
Now the very first thing is that we like to try to answer is how does it get internalized? Does it release effectors? Because we are still trying to figure out how is it entering. And in macrophages, a lot of mechanisms have been blocked. Upon internalization, the autophagy, the ribosome, everything has been blocked. So we have to activate them, but in MSCs we are trying to look at it. So once we can, if we can decipher that, then I'll be able to answer your question a little better.
Okay.
Is it in therapeutic potential? Is it therapy? It's clearance, or
I don't know, ma'am. I'm sorry, I didn't.
Is it that it's performing a clearance mechanism, or what is
DPSCs?
Yes.
So far, no. They have been harboring the pathogens within the host cell, but we have seen post 96 hours, this data has not been shown, where the cells have been opened and the pathogens try to escape outside. Okay. So the media that I maintain, these infected cells are in 10% plus gentamicin. So this gentamicin is given so that the pathogen doesn't escape. But because of overcrowding, that right now at a higher load, they try to burst out from the cell, and there is cell death as well as, so we are still trying to look at. That's where our question comes, can we use rapamycin to turn on any pathways and kind of bring down the clearance? I think I'm the lucky one.
Thank you, Mr. Allen, for the wonderful presentation. Since time is a bit on the short side, I request the audience to keep the questions to a max, like 2 questions for each presentation. For the next presentation, I call upon Ms. Tejasri Wanje.
So good afternoon, everyone. My name is Tejasri. I'm a third year PhD student. I'm working under the guidance of Dr. Shagufta Parveen. So the PhD that I'm working on is entitled as Modeling Neural Tube Defects Using iPSC-Derived Neural Organoids. So neural tube defects are severe congenital malformations that result in failure of neural tube closure. This happens during the embryogenesis, and the prevalence of neural tube defects is 2 per live births, 2 per 1,000 births, with high prevalence in stillborn and pre-viable births than among live births. So depending on the area and the skin covering over it, the neural tube defects are classified as anencephaly, encephalocele, craniorhizoschisis, and spina bifida. The anencephaly is a total or partial loss of brain and the cranial vault. Encephalocele is a sessile cystic lesion which protrudes out through the skull. And craniorhexis is a combination of anencephaly adjoining by the adjoining bony defects of the cervical spine. And spina bifida is a bony defect of posterior vertebral arches, which is accompanied by herniated neural tissue. So development of neural tube occurs in the 3rd week of embryogenesis. Here I have a small video which would help you understand how the neural tube develops. So this is a neural plate. These are the neuroepithelial cells. The neuroepithelial cells undergo apicobasal constriction, due to which there is formation of a neural groove, which then elevates, and this groove fuses in the center, resulting in formation of a neural tube. After the fusion, there are neural crest cells that start appear and migrate out, and the neural tube then separates out from the surface ectoderm. So I'm studying the neural tube development, for which I'm using 3 different model systems. For example, for the first one, I'm using the iPSCs, which are corresponding to the stage of blastulation, and these iPSCs are equivalent to the inner cell mass cells. Then followed by neuroepithelial-like cells, which mimic the early neural tube development, and followed by neuroepithelial-like organoids, which are 3D structures of early neural tube development. So neural tube defects are considered to have a multifactorial etiology, which includes genetic and environmental factors. So dysregulated epigenetics is particularly during the embryogenesis, results in genome-wide methylation, which has been linked to neural tube defects. And the maternal exposure to various teratogens like valproic acid, azacitidine, and dietary folic deficiencies lead to neural tube defects. So coming to the azacitidine, it is an cytidine mimic wherein one carbon is replaced by nitrogen, and this azacitidine covalently bonds to DNMT, which results in inhibition of DNMT gene. So depending on the concentration, wherein high doses causes the cell death, low concentrations of azacitidine results in cell replication without DNMTs. And then I'm looking at folic acid, which is already known to rescue neural tube defects, but the underlying mechanism, how it rescues neural tube defects, is not understood. So for which, as I had told earlier, that I've used iPSCs, which are equivalent to the inner cell mass. I have cultured the cells in 0.5 micromolar of azacitidine, where I've seen DNMT3B was significantly downregulated, and by addition of folic acid to the cells, there was rescue in DNMT3B. And the other pluripotency markers and proliferation and all was not significantly up or downregulated. Then I've generated the neuroepithelial-like cells, in which also I could see there was a downregulation of DNMT3B and rescue of it by addition of folic acid. And the TET2 was also going up, but it was not getting rescued by addition of the folic acid. Similarly, I could see that PAC6 and SOX2, which are neural stem cells marker, are significantly upregulated as compared to the control cells. Further, to model it in a 3D aspect, I have generated the neuroepithelial organoids, for which I have seeded single-cell iPSCs in 100% matrigel and cultured them for 6 days in N2B27 media. So here, I have cultured the cells in normal control media, low folic acid condition, 0.5 azacitidine and 0.5 azacitidine and folic acid. So when we did a quantification with the sizes of control and low folic acid condition, I've seen that the low folic acid cysts were comparatively very smaller as compared to the control conditions. So basically this cyst in the center, they have a lumen-like structure, and surrounding to which are pseudostratified neuroepithelial-like cells. So these are mimicking the early neural tube development. When I did the quantification for 0.5 aza and folic acid, there was also a significant increase in the size when I added the folic acid, and the viability was also high when I added the folic acid. So I have done a IF for SOX1 and nestin, which is a early neural stem cell marker. As you can see here, as compared to the control, 0.5 aza cyst, you can see immature neurites that are staining positive for nestin as compared to the control, wherein the neurite projection is not seen. And with aspect to low folic acid, it was similar to the control. only the difference seen between the low folic acid and control conditions was the size was relatively very smaller. When I added the folic acid to the azacitidine treated cyst, there was a little bit rescue, or not rescue, but the neurites were not seen as many as seen in 0.5 aza conditions. So I have done a beta-3 tubulin and N-cadherin staining. Beta-3 tubulin is a matured marker, matured neural marker. So compared to the control, 0.5 aza and folic acid, you can see that there is thick bundles of beta-3 tubulin expression seen. So when I did a staining for OCT4 and NANOC, which are pluripotency markers, I could see some amount of expression in the control and low folic acid condition, but in the 0.5 aza and folic acid, the expression was relatively lesser as compared to the control conditions. And then I've done a staining for a podocalixin, which would tell me that the center, whatever lumen or cavity is formed, it's not an artifact, but it is actually a lumen. So in the center, you can see the green, that says that it is a lumen and not an artifact. So with 0.5 aza and folic acid, the lumen was not in the center, but towards the periphery. Yet I will not be able to tell right now that if it is an abnormality or it is something that it is showing up in the cells. So I've done again a staining for ezrine and ZO1. So ZO1 staining basically I've done here to check if the apico-basal polarity is forming with the cells or not. So that I could check by seeing the constriction of the ZO1 staining, which is projecting towards the lumen, suggesting that the apico-basal polarity is established with the cells and with also 0.5 aza and folic acid. So to summarize all this until here, what I think azacitidine is doing is it's inhibiting DNMT3B, which is resulting in either global or gene-specific hypomethylation, which is causing enhanced in neurogenesis, which could be seen with the cyst forming the neurites, which are positive for nestin, and the formation of thick beta-3 tubulins, which is saying that the neurogenesis is enhanced. Similarly, with the neuroepithelial-like cells also, the PAX6 and SOX2 was upregulated, which proves the same point. I've also generated neural trunk-like structures to mimic the anterior basal polarity and formation of the neural tube, along with the somites to model the co-development of neural tube and the somatogenesis. So this here you can see X and Y axis, wherein in the bottom picture you can see a circle that, and in the center you can see a lumen. That means that it is forming an anterior-posterior neural tube. So this model system I will use and treat this model with azacitidine and see if at all there is any difference or anything that I've observed with the cyst. Will I be able to see the same with this? Now I would like to talk about encephalocele in specific. So encephalocele is a sessile cystic lesion that protrudes out through the defect in the cranium. So the prognosis of this defect depends on the size, location, and the amount of herniated tissue that is seen. So based on location, encephalocele is also of different types, of which occipital, that is present in the posterior of the brain, is more prevalent. So I have received a human fetal sample. In the first picture, as you can see, there is a sac-like structure that is present at the posterior of the skull. That is basically an occipital encephalocele. So I've isolated the MSCs and dermal fibroblasts from this patient cell, and I've reprogrammed them to iPSCs. So for the interest of my study, I am using iPSCs which I have generated from dermal cell fibroblasts. So as a part of characterizing the cells, I've done a karyotypic and STR analysis. Okay, I'm almost done. So these lines I've done all the characterizations for trilineage differentiation and this pluripotency markers. So basically I can say that I have an iPSC cell line which is specific to the encephalocele human neural tube defect. And I've also done a whole genome sequencing of this sample, wherein I've used the parent's blood and the tissue sample that I've received from the patient. And the analysis part is yet to be done, but there was a copy number variant that struck us was FAHD2B, which is a fumarate acetohydrolases, which is involved with the oxaloacetate metabolism. So I am interested specifically to look into this because this is also hinting us towards the folic acid metabolism. These are all methylation patterns that we have checked, but we are yet to analyze this data completely. So further, I will be using this iPSCs derived from the patient samples and use this for studying this. Thank you.
Only student asks 2 questions, not more than that. Okay, no questions. Next.
Thank you.
Thank you, Ms. Tejasri, for the wonderful presentation. Next, I invite Ms. Rajasri onto the stage for her presentation.
Good afternoon all. Myself Rajashree. I am doing PhD under the guidance of Dr. Vinay. My topic of a talk is epigenomic consequence of inflammatory cytokine signaling in cachectic muscles. Cancer cachexia is a multifactorial syndrome in which patient suffers from a significant weight loss which is irreversible, and it largely affects skeletal muscles. The cancer patients, like 50 to 80% cancer patients develop cachexia because there are no robust biomarkers and approved drug therapies available to cure the conditions, and the underlying mechanisms through which skeletal muscle loss takes place is not clearly explored. This condition is mediated by various factors. For normal muscle to grow properly, there should be balance between protein synthesis and protein degradation. But in tumor conditions, tumor releases certain factors like inflammatory cytokine and growth factors and hormones, and all these pathways converge into the activation of E3 ligases, MAF, BX, and MIRM1, which leads to more protein degradation. But along with that, there is an inhibition in the expression of MyoD, which is a master regulator of myogenic differentiation. So apart from this tumor released factors, there are other negative regulators which are playing role. One such known negative regulator of myogenic differentiation is G9A, and in this project we are trying to understand, does G9A has a role in cachexia. So what we took human skeletal muscle tissues from human patients, control patients, cancer cachectic and cancer patients, and we found that the levels of G9A were elevated in cachectic patients correlated with phosphostatin 3. Phosphostatin 3 is a downstream protein of IL-6 signaling, and for next we wanted to understand, does its mRNA levels also increase? So we did mRNA expression analysis, and we found that the mRNA of G9A doesn't increase in these patients. There is no change. So next we wanted to understand this particular thing. So we took C2, C12 myoblasts as a culture system to work, and we treated them with 25 nanogram per ml concentration of IL-6, and we found that consistent with our human patient data, the G9A levels protein levels were getting gradually, they were gradually increasing correlated with phosphostatin 3. Then we wanted to check its mRNA expression, and we found that its mRNA expression does not change. So next we wanted to understand, does as like protein levels are high, but mRNA does not change. Does this protein is getting stabilized? So we performed cyclohexamide chase assay. We overexpressed C2C12 cells with GFP tagged G9A and treated them with 50 nanogram per ml concentration of CHX along with Mg130 to 10 micromolar per ml concentration and IL-6, and we found that in presence of CHX, protein degradation was taking place. CHX is but in presence of IL-6, the protein degradation was rescued, similar to Mg132. Mg132 is a proteasomal inhibitor. So from that, we concluded that in presence of IL-6, G9A protein is getting stabilized, and generally it gets degraded via proteasomal degradation pathway. So after that, we wanted to understand, does G9A gets ubiquitinated as it gets degraded via proteasomal degradation pathway? So we did immunoprecipitation, and we found that in presence of IL-6, G9A is getting less ubiquitinated compared to control. Further, now it is getting, generally G9A gets ubiquitinated, but in presence of IL-6, it is getting less deubiquitinated. There must be some molecule which is deubiquitinating this G9A. So there is a group of enzymes called as deubiquitinases. They deubiquitinates the protein. We did literature review, and we found that in presence of IL-6, there is a one DUB3, deubiquitinating enzymes, that is DUB3, which gets upregulated. So we wanted to understand, does there is any interaction or correlation with G9A? We performed string analysis, and we found that there is some connection between IL-6, G9A, and DUB3. So what we did, we treated C2C12 cells with IL-6, and we wanted to check for the mRNA levels of DUB3, and we found that in presence of IL-6, DUB3 mRNA levels get increased. So further, what we did, we overexpressed GFP tagged DUB3 in C2C12 cells, and we wanted to understand what happens to G9A levels. So in presence of overexpressed DUB3, G9A protein levels were elevating. Further, now from this, we wanted to check, does G9A interacts with DUB3 and making it stabilized in presence of IL-6. So we performed co-immunoprecipitation study, and we overexpressed cells with GFP tagged DUB3 and flag tagged G9A. And if you can see, there is a interaction between G9A and DUB3. So from overall, we concluded that in presence of IL-6, DUB3 interacts with G9A and makes it more stable. So next we wanted to understand what happens to the differentiation, what is the effect on differentiation. So we differentiated C2C12 cells for 2 days, and we checked for the levels of myogenin and troponin T. Myogenin and troponin T are the myogenic regulatory factors, and we found that in presence of IL-6, the levels of myogenin and troponin T were down-regulated. So next we wanted to understand what if we inhibit the activity of G9A, what will happen? So we used UNC0642, which is a potent inhibitor of G9A's activity, and we found that in presence of UNC0642 and IL-6, the expression of protein levels of myoG and troponin T were rescued. Next, we performed immunofluorescence assay. We differentiated C2C12 myoblast for 2 days, and we treated them with IL-6 and IL-6 plus UNC0642, and we found that in presence of IL-6, there is less differentiation, but when we treat them in combination with UNC0642, the differentiation was rescued. Next, we wanted to understand, does G9A gets less ubiquitinated in presence of IL-6? So we performed co-immunoprecipitation using differentiated cells, and we found that in presence of IL-6, G9A was less differentiated at day 1. So from all this study, here we conclude that there is a crosstalk between inflammation and epigenetics in cachectic muscles. generally in normal muscle differentiation, G9A gets proteasomally degraded, which leads to activation of muscle promoter and transcription of myogenic genes, leading to proper myogenic differentiation. But in case of cachexia condition or impaired myogenic differentiation, increased levels of IL-6 stabilizes DUB3, increases DUB3, and this DUB3 stabilizes G9A by rescuing it from its proteasomal degradation, and there is inactive transcription of muscle promoter leading to impaired myogenic differentiation. Thank you. For now, I'm mentioning just specific ubiquitination because I haven't checked for it, like if it gets polyubiquitinated or monoubiquitinated.
Also, whether it's getting case 63 ubiquitinated or
No,
or
I haven't
40.
checked it.
Oh.
I'll look into it.
Okay.
Yeah, we have, apart from IL-6, I have also used TNF-alpha, but it doesn't give us the same thing like IL-6. G9 expression does not change in presence of TNF-alpha. Because of time constraint, I didn't put the data. So, IL-6, right?
And the dub that you
DUB3. So there are reports, they suggest that in IL-6 induces the expression of DUB3. It's like there are paper published.
like a stat target?
That 100% I don't know, but yes, like the report says that in presence of IL-6, it's upregulated.
But if it's a direct or indirect, you don't.
That I have to check. Yes.
It's a stat target. Oh, yeah.
Thank you, Ms. Rajashree, for the wonderful presentation. Now I call upon Ms. Aishwarya Raj for the next presentation. You want to use the is this okay? Yeah, it's fine.
Okay. Good afternoon all. I'm Aishwarya Raj, second year PhD student from Dr. Urmi's lab, and today I'll be talking about a part of my study which is titled as non-canonical role of RAG GTPase in cellular remodeling under pathophysiological conditions. When cells are growing in a nutrient-rich environment, there is a localization of the mechanistic target of rapamycin to the lysosomal membrane with the help of the RAG regulator complexes. Can I have a pointer? Pointer doesn't work. Okay. I hope you guys can see the mTOR and the RAG regulator complexes there. So with the help of the RAG regulator complexes, the mTOR localizes to the lysosomal membrane where it initiates the downstream anabolic responses. From our lab, the previous study has shown that in the leucine deplete environment, there is a storage of leucine that is happening within the lysosome. The interesting part of this study was that this particular storage was independent of This particular storage was independent of the activity of the mTOR kinase, which makes it different from the canonical pathway that I have just mentioned. And they have tested in a different cell lines. Across all cell lines, this particular pathway was conserved, except for one cell line that was the MCF7 breast cancer line. In this particular cell line, upon investigation, it was found that the cell line is not capable of storing leucine within their lysosomes, even in a leucine-deprived environment. And to further know why this particular thing happens, they found that there is a downregulation of RAG GTPases in this cell line compared to the breast epithelial cell line. Oh. It's not moving. System is hanged. Okay, yeah. Sorry, I didn't know the slide was not there. So this is the MCF7 condition that I was mentioning, where they have a low RAG GTPase level when compared to the breast epithelial cell line. And when we overexpressed the RAG GTPases in this cell line, there was a rescue of the phenotype, the overexpression phenotype. But what piqued my interest from there was that these cell lines had a tendency to grow in a different morphology when compared to their wild type MCF7, where the MCF7 cells grow more like a 3D spheroid-like structure, while the MCF7 with the overexpression had a 2D monolayer-like morphology. Now, to understand why this kind of a pattern change happened, in literature it is known that the intermediate filament, vimentin, can be one of the candidates which can change the cytoskeletal and the cell-cell adhesion pattern. So we looked into the cytoskeletal marker vimentin levels in this cell line, and we could see that there is a lowering of vimentin in this overexpression scenario, suggesting there is a translational downregulation of vimentin upon RAG GTPase overexpression. Now, is it just a scenario that is seen in the MCF7? So to understand that, we looked into multiple other cell lines, and we could see that in other cell lines also, there is a correlation that is happening between the RAG GTPase level and the vimentin level, where when you have a high RAG GTPase, there is a lowering of vimentin, and when there is a low RAG level, you have a high RAG GTPase. To confirm this further, we looked into a 3D-like scenario where we grew these cells in a ultra-low attachment plate. And we could see that when you make these cells more 3D, they are losing their RAG levels, and inversely, there is an increase in the vimentin levels, suggesting that there is a similar morphological and molecular machinery that is acting in order to maintain this relation between the RAG and the vimentin level. Now, to get into a more in vivo-like condition, we looked into these cell lines in a matrigel, and we could see that when these cell lines are grown in a matrigel, the spheroids that they form were very different, where MCF7s had the tendency to have this projections coming out of their periphery, while the RAG overexpression lines, as you can see, is very compact with a well-defined margins. This made us wonder if there could be a change in the EMT marker expression as such, and we looked into the other markers other than the vimentin, and we could see that there was a clear reduction in the mesenchymal markers, and there was an increase in the epithelial markers, suggesting that the RAG overexpression is somehow suppressing the mesenchymal property and enhancing the epithelial property. Now, is it just at the translational level, or is it at the transcriptional level as well? So to answer this question, we had a RNA-seq analysis done, and we could see that upon overexpression, there is a change in many pathways, and one of these being the cell adhesion and the extracellular structural organization pathway. And among them, we could see that there were many mesenchymal and the epithelial marker changes, as we have seen in our qPCR also, suggesting there is a transcriptional rewiring of these marker genes happening. Now, something else also caught our attention in this particular RNA-seq result, that was the autophagy gene regulation. There was a downregulation of many of the autophagy-related genes, and we wanted to know if there is a change in autophagy as such. So we looked at one of the functional assay for autophagy, that is the autophagy flux assay, by looking at the LC32 turnover. So we could see that when the RAG levels are high, there is a lowering of the autophagy flux, suggesting that with the autophagy gene downregulation, there is a autophagy functionality loss as well. Now, to confirm what we have seen with the autophagy functionality loss, we did a knockdown of these autophagy genes, that is the macroautophagy genes ATG3 and ATG13, and we could recapitulate a RAG-AB GTPase overexpression-like phenotype, where with the downregulation of ATG genes, we could see a downregulation of vimentin. This got us to this hypothetical model where RAG-AB GTPase regulates the autophagy, where it down to the autophagy, the vimentin is downregulated, ultimately giving a cellular growth pattern difference. But when I talk about the RAG-AB GTPase overexpression, we had one more concern in our mind, and this was that if I'm overexpressing RAG, there could be activation of mTOR as such. So we wanted to know if we are changing the mTOR activity, and one way of looking at it is by looking at the phosphorylation state of the mTOR substrates, that is the S6K and the 4EBP. Interestingly, we could see that there was no much change, or there was no significant change in the mTOR activity as that is noted by the phosphorylation state of these substrates, which puts us in this scenario where we wanted to look if the vimentin level or ecadherin level will change if we inhibit the mTOR activity by putting a drug called as taurin. So we added a taurin drug, and we saw that even in the presence of taurin, this particular change that we observed for the vimentin, ECAD, and the mesenchymal markers were retained, suggesting whatever changes that we are observing with the RAG overexpression is an mTOR independent process, which puts us in a non-canonical scenario as compared to the canonical mTOR dependent pathways. So RAG GTPases, as I have mentioned before, for a long time was considered to be dependent to function with the mTOR pathway. It was always correlated with the mTOR activity and mTOR activation amino acid sensing. But recently, the studies have shifted a little bit to the mTOR independent role of these GTPases. And we, from our lab also, as I have mentioned in the beginning, have contributed to that study where there is a lysosomal amino acid storage homeostasis happening. Now, I would like to add one more layer to this independent function where we are seeing a cytoskeletal remodeling. And as we know, in a physiological scenario, the cytoskeletal remodeling could give rise to many other features we could explore in the future, which includes the embryonic developmental stage or wound healing, etc. At the end, I would like to thank all the members of my lab for helping me with this project, my guide Urmi, and Dean, my DAC members and collaborators who have guided me and who have given me all the suggestions for this project. Thank you. Mesenchymal to epithelial. That is what we are thinking with the protein.
Epithelial and maybe metastatic.
That's what, with the protein levels, that's what we are seeing. There is a shift in their mesenchymal property to an epithelial property. We need to further explore, but right now we are not trying to look at it from a cancer perspective, but more in a generic phenomena.
Okay. And can this be extrapolated to other than MCF7, or is it restricted to?
We have looked at the different cell lines from the RAG and the vimentin axis, and we have seen that inverse correlation in all these cell lines. When there is high RAG, there is a low. And the spheroid that I have shown is a HEK cell line, which is not even cancer cell line.
That's what I was thinking.
I think there is this relation between the RAG and the vimentin is maybe we can extrapolate it to, that's why I was suggesting maybe in the future we can look into embryonic development or any other scenarios.
Okay.
Something?
With the RAG, RAG can regulate the lysosomal pH, but in this MCF scenario, I haven't looked into the pH as such. But yes, RAG can regulate the lysosomal pH.
Right.
And
no,
the other
because if there is lower pH differences, possibly the engulfment of into the lysosome and would vary definitely.
The storage phenomena is restored, but in a parallel study in our lab, we have seen that when there is a storage, there could be an alkalinization of the lysosome as well. So that could be there, but in the MCF7, we haven't looked at the alkalinization or anything.
Right, thank you.
Yeah, thank you. You will close the
Thank you, Mrs. Aishwarya, for sharing your valuable insights with us. Next, I call upon Ms. Janani Yuvaraj for the next presentation.
Good afternoon, everyone. Myself Janani, and I am JRF under Dr. Shweta Chakrabarti. So I'll be talking about Drosophila innate immune priming to various pathogens. To start with, Drosophila, which is also known as fruit flies, spend most of their lifetime in decaying fruits and vegetables. And we all know the rotting fruits has a lots of pathogens such as fungus, virus, bacteria, and even wasps. So day-to-day life, the flies are getting exposed to multiple pathogens. So how is it able to shape its Yeah. So I was explaining the left diagram. Day-to-day life, they are getting exposed to multiple pathogens. So how are they able to shape their immune system and able to live better? So that is one of the important point to be considered. So knowing about the general mechanism, when there is a microbial entry attempt, Drosophila has a very good physical barrier such as tough cuticle, acidic pH, and even gut enzymes such as lysozymes. So these help in preventing the microbial entry. But what if chance the microbes breach into the flies? So two types of immune systems or immunity can be activated, the humoral response and the cellular response. Humoral response is mediated by fat body, the fly analog of liver, and involves systemic production of antimicrobial peptides. The other response cellular involves immune cells called hemocytes, which involves three cell types: plasmatocytes, lamellocyte, and crystal cell, which are equivalent to macrophage, natural killer cell, and granulocyte. So they produce toxic quinones and hydrogen peroxide that helps in killing the bacteria. It is changing here, but not changing there. Yeah. Now there are also multiple studies being conducted on analyzing what are the pathogens that are associated with the flies. One such example is Enterococcus faecalis, which is a gram-positive bacteria and was isolated initially from the gut and hemolymph of the wild Drosophila. Second one is Erwinia keratovora 15, which is a natural pathogen for the fly, and it is a gram-negative, and it is also a phytopathogen. So it causes soft rot disease in plants and vegetables. It uses Drosophila as a vector to spread disease among the plant population. Third one is Candida albicans, which is a class of fungus and forms hyphae and filaments, and it is associated with causing, with its virulence and activating the immune response in flies. And everyone must be familiar with the difference between gram-positive and gram-negative, but just to highlight, gram-positive has a very thick peptidoglycan layer, and the negative has very thin peptidoglycan layer coated with lipopolysaccharide layer. Yeah. So now we are exactly moving to understand what is innate immune priming. You all must be familiar with immunization, which involves adaptive immunity. And one good approach to protect human diseases is identification of vaccination. So that happens through T cell and B cell. But there are growing evidences suggesting not alone adaptive immunity, but also innate immunity is playing a key role in immunological memory. Because we all have been grown up saying that only adaptive has immune memory, not the innate. So there are recent literature suggesting that immune memory has been identified in spiders, scorpions, mosquito, and flower beetle, and many other invertebrates. And one live example of innate immune priming and memory is BCG vaccine. We all know BCG vaccine is used for curing tuberculosis, but recent evidence and recent articles suggest that it is also involved in protecting from other non-related infectious disease and some cases cancer as well. What is the mechanism associated with? Like, studies are ongoing, but they aren't sure what is the real exact mechanism associated. From this diagram, we know that immune responses, first we have initial primary challenge, which involves attenuated pathogen. We have first response, which is less and able to produce antibodies. The second infection, the secondary exposure, includes a very heightened and a faster response, which we term as trained response. And many papers suggest that this could be due to some metabolic reprogramming, epigenetic modification, and other histone modification. It's not sure, they are still working on it. And the important note for today's thing is the background, where a study conducted by Shweta and Sandhya, they showed that prior injury is able to confer protection against a subsequent gram-positive E. faecalis infection. So the study is of MAHE fly categorized into two, unchallenged and clean injured. Unchallenged, no prior exposure, directly getting exposed to bacteria. Clean injured is priorly injured, 2 days post-infection, they are getting exposed. And you can see a clear difference in the survival. And they specifically saw this heightened antimicrobial peptide response in hemocytes 20 hours post-infection. So this led to hypothesize that does this injury-induced protection seen only in gram-positive, or it can also be seen in gram-negative and other pathogens such as fungus. The exact experimental paradigm was followed. So everything is exact, only the gram-positive bacteria was replaced with the gram-negative bacteria, and we are seeing the same significance in the survival. The primed individual were able to survive better when compared to their naive counterparts. So this difference in survival, does it mean the fly was able to kill the bacteria? Yeah, because since there is a difference, it should be able to kill the bacteria. But the bacterial load analysis, the CFU result says no difference, no significant difference between the two groups. So what does that exactly mean? The flies were able to tolerate the pathogen and survive better. So through what proper mechanism is the protection induced? Is it tolerance? Now you guys, many must be familiar, many must not be familiar. What is tolerance and resistance in fly immunity? So the relationship between the host and the pathogen can be represented by a tolerance curve. Increased tolerance means the flies are able to withstand the bacterial load without changing the load and improve the health of the fly. Resistance is reducing the pathogen, killing the pathogen and surviving better and improving the host health. And there are multiple studies that are ongoing with tolerance and resistance. As I mentioned early, there need to be multi-facet studies in immunity, not just survival, and 2 studies can conclude. One such important study is melanization. So melanization in flies involves immediately after wounding, we have a cascade of serine protease reactions happening that activates a melanization enzyme, important melanization enzyme, that leads to rapid deposition of melanin at the site of wound. So this melanin deposition is one of the key immune response in the flies. Here we are scoring melanization in flies into different categories, like you can see from the image, light, medium, and dark. And we observed the injured group had less melanization intensity when compared to their unchallenged part. So what is the thing happening between the unchallenged and the injured group that the melanization is able to reduce? So now we moved on to another different pathogen, which is fungus, Candida albicans. Again, exactly everything is the same, and you can see the difference in the survival. Unlike ECC15, which had a good significant difference, this is even drastic, like similar to E. faecalis, what we observed priorly. And again, we asked question, is this through resistance or tolerance? Again, the bacterial load is saying there is no difference between the unchallenged and the clean injured. So is really tolerance explaining the protection that is provided by the injury? We have to work more and find out. And same, One more important thing to be highlighted here is we saw 3 type of intensity with a less pathogenic ECC15 infection. But here, since Candida albicans is very highly pathogenic to the flies, you can see one more extra melanization phenotype, which is robust. And there is a really clear significant difference between the unchallenged and the clean injured group when looking at their melanization. So is this reduced melanization correlates to improved survival? We have to work on it. So to summarize, E. faecalis, which is a gram-positive bacterium, Candida albicans, which is a fungus, and ECC15, which is a gram-negative bacteria, everything goes with a very distinct pathway. Like E. faecalis and Candida goes through toll, and ECC goes through IMD. But how with this different pathway, they are able to confer protection when primed. So this raises a question, could melanization be the link in conferring the protection? And is this the common link? Because as I mentioned earlier in the melanization scheme, one of the metabolic intermediate is H2O2, which is ROS, that is able to kill the microbes. So this increased ROS, is it leading to immunopathology and leading to collateral damage? We aren't sure. So future plan is, we now phenotypically saw how melanization is there. Now it's to quantify using L-DOPA and analyze the melanization pathway. One more thing is to genetic approach of analyzing the survival with flies that are mutated with melanization. So without melanization, is protection there or not? Third one is to quantify the bacterial load in E. faecalis, because we saw for ECC15 and Candida, whereas E. faecalis priorly it was not done, so that will also be done. And next, does the route of infection causes any difference? So far we saw is sepsis to systemic infection. So does the local wound through oral infection? There are 2 modes, systemic and oral. So does oral leads to any difference, or it also confers protection? We have to work on it. Thank you, and we thank MAHE and DBT Welcome India Trust for funding our study. Thank you.
Thank you, Miss Janani, for the excellent and thank you, Ms. Janani, for the excellent and informative talk. Next, we will have our last presentation, which is by Ms. Prerna Singh.
Hello. For this, hello. Fine. I'll speak louder than this. Okay. This, right? Yeah. Okay. Thank you. The light should be on. Call it. Hello. Imagine you're a cell under stress. Can be oxidative stress, can be starvation. You need to make a crucial decision to stop all the processes and protect what matters the most. Today I'm going to talk about how a cell makes this decision through specialized structures called stress granules. And I'm also going to talk about a protein we usually relate to translational machinery, but how it can be a protein that regulates the cellular homeostasis and also can cause disease, might cause a disease. Good afternoon, everyone. I am Prerna Singh, working in Dr. Amanjot's lab. And today I'm going to present our lab's work, and which is so the main focus of our lab is to study the protein aggregation and how the dynamics of it and how it can cause diseases. So protein aggregation is a general feature across neurodegenerative diseases. In Alzheimer's disease, we have tau and amyloid beta aggregates. In Parkinson's, alpha-synuclein make Lewy body. And ALS and FLD are characterized by TDP43 and FUS aggregates. What we need to understand is that not all aggregates are pathological. In this video, I show that the C. elegans P granule is crucial when the cell is going through embryogenesis. These are dynamic structures and are dynamic and functional structures which are required during embryogenesis in C. elegans. We have different type of aggregates which are functional. Germline granule, which I showed right now in C. elegans. Neuronal granules, which help in local translation at synapses. Nuclear granules, which are formed in the nucleus and for transcription, and there are stress granules. To understand how these stress granules are formed and what is the difference between the stress granules, the pathological aggregates, and the normal aggregates, we need to understand the integrated stress response. So EIF2 alpha is the initiation factor of translation, and during various stress conditions like viral infection, ER stress, this EIF2 alpha is phosphorylated at serine 51, which leads to the shutdown of cap-dependent translation, and eventually it leads to stress granule formation. Here we have a video showing how dynamic these structures are, and here in this video, G3BP, which is a core stress granule protein, is tagged with M-cherry. Thanks to Ashish, Mithun, Sanjana, and Chandan in our lab for this beautiful data. And we can see here in this video that in 30 minutes of sodium arsenite, which is oxidative stress, we can form stress granules. And to show how dynamic they are, we here have ataxin 2, which is also a marker of stress granule. In stress conditions, these are formed, and then these get resolved as well. The dynamicity of these granules is important, because if these stress granules persist and they don't disassemble, it will form a persistent granule which can be pathological. So a study shows that, and these stress granules are mainly composed of ribonucleoproteins, mRNA, which are not translated, and RNA binding proteins. And proteins, these proteins are especially proteins which have intrinsically disordered region. You can have multiple types of disordered region, like alpha-synuclein is a completely disordered protein. You can have a disordered tail, a disordered loop, and disordered linker, which connects two ordered domains. So we wanted to know what these stress granules consist of. So we did immunoprecipitation with ataxin 2, followed by a mass spectrometry, and then a proteomic analysis. So we found the usual suspects you would expect in a stress granule proteome. What interests us here is the AIMPs and the tRNA synthetases. I would like to tell you that these AIMPs are not just random proteins. These are translational machinery which charges the tRNAs with its cognate amino acids. And it's called a Mars complex, which is a multi-amino acyl tRNA synthetase complex, which consists of 8 tRNA synthetases and 3 scaffolding proteins, which are AIMP1, AIMP2, and AIMP3. These proteins basically perform the role of scaffolding this complex, but more than that, these AIMP2 also has a disordered region. And AIMP2 specifically is the target for parkin, which is a E3 ubiquitin ligase, which is known to be mutated. The parkin is known to be mutated in the familial Parkinson's disease. So to check if our data from the previous mass spectrometry, we checked that the co-localization of AIMP2 in control and stress conditions. In stress conditions, we saw that AIMP2 is co-localizing with G3BP1, which is a core stress granule protein, which confirmed that AIMP2 is recruited in the stress granules. So knowing that AIMP2 has a disordered region, we wanted to check that if it has intrinsically aggregating property due to its disordered region. So we overexpressed AIMP2 in multiple cell types, and we found that without any stress, these aggregates are formed upon overexpression of AIMP2. And what is interesting here is that these aggregates are formed even in the absence of G3PP1 or 2, in what we saw in G3PP1-2 double knockout cell lines. So, and we wanted to check if these aggregates are fluid-like or solid. So we did a 1,6-hexane diol experiment, which breaks weak hydrophobic bonds. And we see some difference in the wild type and the G3BP knockout aggregates, but we are still testing this. And if you ask me what other components are there in these aggregates, we stained it with ataxin 2, AIMP3, which is again a Mars complex. And the presence of ubiquitin might suggest that these aggregates are aggregating the ubiquitin, the proteasomal degradation machinery as well. So where does this leave us? So we know that EIF2 alpha is phosphorylated in stress conditions, which blocks the cap-dependent translation at the initiation. But AIMP2 can it be another way of the cell to as a protection mechanism to block translation, not at the initiation step, but at elongation. This can also, we have another question here, what can be the difference in the translation regulation when we have AIMP2 as a condensate and when we have it as a soluble form, and how this is going to affect the translation, overall translation in the cell. I would like to thank my lab members, and no amount of thanks will be complete without acknowledging the core protein of the aggregate we have here, a small aggregate we have here in our lab, Dr. Amanjot Singh. Thank you for all your guidance, your patience in teaching us. Thank you. And thank you, Manipal, for providing me with the platform. Thank you.
Thank you, scholars, for the wonderful presentation. An institution's true pride lies in the achievements of its alumni. Now, it is my privilege to invite Dr. Raghu Ram, one of our alumni, whose journey stands as an inspiration to the current scholars. So, we are honored to have you here with us. Kindly address the gathering.
Good afternoon, respected faculty, distinguished guests, and students. My name is Dr. Raghuram Bhandari, and I currently serve as the CEO of Nepal Stem Cell Research Center. It is a real honor to be here today, not returning to MIRM, not just as a guest speaker, but as an alumnus. This institution played an important role in shaping how I think about science and research. I thought the best way to share my journey would be through three parts: the past, the present, and the future. I think I was always curious about life. One of my favorite childhood books was Bones, Bones, Dinosaur Bones. While other kids were fascinated with superheroes, I was fascinated by fossils and the history of life. In high school, my science project was building a miniature biosphere, a small ecosystem inside a glass container. Looking back, I think that project reflected something about me. I wanted to understand life. I wanted to understand how life works as a system. That curiosity took me to the Philippines for medical school. After graduating, I returned to Nepal and chose to begin my career at the foothills of Mount Everest, serving communities where access to healthcare is limited. That experience grounded me early in my career and reminded me that medicine is not just science, it is responsibility. The turning point in my journey came when I attended my first International Society for Stem Cell Research conference in Seoul, South Korea. For the first time, I saw scientists thinking about disease not just at the organ level, but at the cellular level. That curiosity eventually brought me to MAHE's PG diploma in regenerative medicine, biology and stem cell technology at the Manipal Institute of Regenerative Medicine. During my time here, I had the opportunity to work on a research project under the guidance of Dr. Uttara Chakrabarti. Working in her lab taught me something extremely important. Science requires structure, discipline, and rigor. MIRM didn't just give me knowledge, it gave me scientific discipline. When I returned to Nepal, I quickly realized something. There were talented doctors and scientists, but very limited research infrastructure. There were a few research centers, but not structured or regulatory framework for stem cell research and therapy. So instead of waiting for something to exist, I decided to build it myself. Yes, I founded the Nepal Stem Cell Research Center, where I currently serve as the CEO. Our goal has been simple: to help create a structured and ethical foundation for regenerative medicine in Nepal. Part of this work has involved engaging with institutions and policymakers to help develop a regulatory framework for stem cell research and therapy, because without structure, meaningful scientific progress cannot happen. Today, our research is working on autologous bone marrow-derived mesenchymal stem cell research, particularly focused on traumatic spinal cord injury. Our work involves harvesting bone marrow, isolating and expanding mesenchymal stem cells, validating their identity and characteristics, ensuring safety and quality standards, and studying their therapeutic potential in both acute and chronic spinal cord injury. This type of work requires collaboration between clinicians, scientists, and research institutions. As we continue building the research ecosystem in Nepal, we are actively looking for academic, scientific collaborations. So I hope today is not only about sharing a story, but also about opening doors for future collaboration. What we are trying to build is not just a single laboratory or a single study. We are trying to build an ecosystem, a place where young scientists can learn laboratory science, regenerative medicine, and translational research. The future I imagine is not just about stem cells. It is about creating an environment where countries like Nepal move from importing innovation to creating innovation. If there is one thing I have learned from this journey, it is curiosity can take you very far. It starts with a simple question, like a child wondering about dinosaur bones, but you continue to follow these questions, and they lead you to unexpected places, from biosphere in a glass, to medicine, to regenerative biology, to building a research center from scratch. And along that journey, MIRM played an important role in shaping how I think, question, and approach problems. So thank you for being a part of the journey, and I hope that some of the curious minds sitting here today will go on to become the people who built what does not exist yet, whether that's in science, medicine, technology, or ideas that can change the world. Thank you.
Thank you, sir, for sharing your experience and valuable guidance with us. May your journey motivate us all in life to aim higher. As we approach the conclusion of this event, gratitude is the finest note on which we can end. I now request Dr. Sweta Chakraborty to present the vote of thanks.
So I have the unfortunate job to hold you from lunch, so I'm going to keep it really short. I prepared something longer, but I'm just going to keep it to a few sentences. First and foremost, thank you, Professor Prasanna and Dr. Meenal, for organizing the National Science Day. Without both of your efforts, this morning, afternoon wouldn't have been possible, so thanks for doing that. Of course, the volunteers, all the student volunteers that you've been seeing on the stage, I won't name the whole list, but they know who they are. Thanks for, running the show and doing it. Our PhD scholars who gave such really invigorating talk, our faculty speaker, Dr. Shagufta, and most importantly, our keynote speaker, Dr. Hemalatha, for, inspiring us to know how you can go from your journey of asking basic fundamental questions to even drug discovery. So that was very enlightening. I just want to leave you with this. Serendipitously, MIRM Science Day wasn't really built around the theme of Women's Day, International Women's Day, which is day after tomorrow, but you can see with 80% representation from, students which are female and women faculties like Shagufta and Professor Hemalatha, I think MIRM is leading by setting examples that, we can have representation of excellent science without having to really think about it. I think we didn't really do it on purpose, but it's the way I think if you just want to represent excellent science, women will come to the forefront. So I won't take much of your time. So thanks everyone for all the participants, and especially from the other colleges, Christ University, Ramaiah, as well as the Karnataka Pharmacy College students to come here and join the National Science Day. And please join us for lunch. Thank you.
Thank you, ma'am. With that, we now conclude this session. Lunch is now served from 1:30 to 2:15. Kindly reassemble by 2. Oh, it's okay. Campus tour would be happening. I hope to see you.